only for Dr. Datta, where we'll have a foundation talk for Dr. Datta. So we'll invite a lot of people. So we
Because
just
there's still a lot of controversies on breast microbiome. No?
Yeah, there are. I mean,
And
it's
your studies would perhaps throw light
I'll
on
ask
it.
Dr. Margaret to just. So
Dr. Nair, this is Tahmid.
he's,
Hi, Tahmid, where are you? Let
Uh,
me see, where are you? You have with the
yeah.
candle in the.
Can you see me?
I can see you, but do you have a brown colored shirt?
No,
it's a blue
it
colored shirt.
is, yeah,
Oh, okay. Yeah, I got you.
almost like yours.
Yeah, I got you.
So how are you? It's so good to see you.
I'm doing very well, Tamil. But I just wanted to congratulate you. What you have done is spectacular. I think it's. Uh, I don't know if Dr. Datta knows Tamil.
No,
Dr.
I don't know him personally, but good morning, Dr. Tamid Ahmed.
Good morning. Good morning, sir.
I'm Saurabh Datta. I'm a neonatologist in PGI, Chandigarh, in India.
Okay. Okay. Very nice to meet you.
Same here, sir.
Dr. Tahmid is now the director of ICDDRB. Tahmid, you're the first Bangladeshi director, no?
That's right. Yes.
Okay, because there were some interims in between, so I was not very clear about
Yes.
that.
Yeah. Dr. Abbas was the deputy executive director, and then for a brief period of time, he was the interim executive director. That's it.
Okay.
Yeah.
But that's spectacular, and I think no one more than you deserve it. I've really
Well,
enjoyed your presentations when we come for the SAC.
no, thank you. Thank you.
And the
Doc-
aspect which I like is your intensity of training of your young colleagues.
Oh, yeah.
A lot
I
of
know
people.
this is something that is, that I like to do very seriously, and, there's a lot of comfort. But Dr. Datta, I must tell you about what Dr. Nair has done for ICDDRP. He's very shy in telling about himself. He was the director of one of the large divisions at ICDDRP for many years. And during his tenure, the Laboratory Sciences Division actually progressed tremendously, not just in terms of capacity building, doing high quality science. The very personality of Dr. Nair, is still an example how, he interacted with the staff, and so that everybody was comfortable working with him. We really cherish those days, Dr. Nair.
Yeah, I really cherish those. Actually, it's I was able to do what I could because of a lot of support from colleagues like you. In fact, I used to
I can
always
fully.
go down your unit downstairs, because I quite liked what was being done.
Yes, I mean, I can fully, imagine and agree with what you're saying about Dr. Nair, because, even
Yeah.
my interactions with him in THSTI, where he was earlier, have been always very pleasant and a
Right.
great source of inspiration, and always smiling. That's what I really like about
Always,
Dr. Nair.
always,
Thank
always.
you. Thank you very much.
Whenever you go to him, it's all positive.
Yes, positive vibes.
Yeah. Thank you, Dr. Nair. And
Oh.
it's so good to see you. I mean, very good health and spirit. So today we start at 10, isn't it?
Yeah, at 10 o'clock.
Yeah. So how much time? It's written 30 minutes. So can I talk for 30 minutes or I end before that?
30 minutes
30
is the
minutes.
time.
Okay.
And then after that, we could have a question answer session.
Okay.
So I had spoken to Dr. Nirja yesterday, So she said around 25 minutes so that we have a little
Okay.
time for questions taken through the
Okay.
chat.
Mhm.
And also in the beginning, a couple of minutes will be spent in introducing the speakers. So
Mhm.
all that put together, 30 minutes.
Okay. So, Dr. Nair, you are now in Delhi. Where are you speaking from now?
I am in Trivandrum, which happens to be
Okay.
my home city.
Okay.
And I
Okay.
have my apartment up here,
Okay.
and my wife and I have
Mhm.
here. I had started some work with the Rajiv Gandhi Center for Biotech,
Sound
Okay.
check.
but I decided not to because I couldn't cope up with it.
Yeah. Yeah. Mhm.
So I decided not to.
Mhm.
But then such occasions come when
Ladies and gentlemen, please be seated. We are going to start this symposium in next 2 minutes. Thank you.
next 2 minutes is precise. Yeah.
Is this also a sit-in symposium where people are seated somewhere? No.
In Delhi.
Oh, okay. Okay.
So it's a hybrid. It's a
Oh,
hybrid.
hybrid. Okay. It's a hybrid. Yeah.
So Dr. Ahmed, do you spend most of your time in Dhaka?
Yes. Yes. I'm based in Dhaka. So before the pandemic, there was a lot of travel, but now,
Yeah, yeah.
nailed down. So which is good. And you are based in Chandigarh.
In Chandigarh.
Okay.
So in fact, my remote
Good morning, ladies
ancestral
and
home was
gentlemen.
Dhaka.
May I have your attention,
Okay.
please?
I see.
A very warm welcome to 11th India Probiotics Symposium. My name is Naina Baveja, formal MC for this symposium. Would like to thank you all for taking out time for participating. And we had overwhelming response at 11th India Probiotics Symposium with 850 people attending this symposium online and 50 who have been present physically. So thank you so much for such a very positive and overwhelming response. So keeping in mind the formal decorum, and we have to proceed with the sequence. May I invite Professor G. Balakrishna Nair and Professor Saurabh Datta to kindly come and to chair this session virtually and kindly introduce the speakers. So I would like to highlight till October 31st, 2018, Professor G. Balakrishna Nair worked in the Research, Policy and Cooperation Unit of the WHO Southeast Asia Regional Office in New Delhi. He then joined the Rajiv Gandhi Center for Biotechnology as an honorary scientist. He was previously the executive director of the Translational Health Science and Technology Institute, Department of Biotechnology. Under his supervision, 29 students have obtained doctoral degrees, and he's the author of over 500 research papers. Everybody, please put your hands together for Professor G. Balakrishna Nair. And I would also like to invite Professor Saurabh Datta. Professor Saurabh Datta is Professor Newborn Unit, Postgraduate Institute of Medical Education and Research, Chandigarh, India. He was faculty member in PGI MER MBBS MD and senior residency from AIMS, Delhi Senior Registrar from the Children's Hospital at West Med, Sydney, FRCPC from Royal College of Physicians and Surgeons of Canada, Education and Research, Chandigarh, India. So ladies and gentlemen, please put your hands together for Professor Saurabh Datta. Requesting both the delegates to chair the session virtually and kindly introduce the speakers. Thank you.
Professor Nair, are you online? And would you be introducing Dr. Tahmid Ahmed? Perhaps Dr. Nair is having some difficulty. He was planning to introduce Dr. Tahmid Ahmed, whom he knows very well personally. So I'll just take over. Dr. Tahmid Ahmed has been working for the last 30 years in simplifying the management of childhood malnutrition, childhood tuberculosis, and diarrheal diseases. He leads the Bangladesh Environmental Enteric Dysfunction Study that attempts to discover non-invasive biomarkers of environmental enteric dysfunction, which is an important cause of stunting in children. And in fact, a major part of his talk is probably going to be on stunting. And together with Dr. Jeffrey Gordon from Washington University, he discovered microbiota-directed complementary foods, a novel intervention for childhood malnutrition. And this discovery was highlighted by the prestigious journal Science as one of the scientific breakthroughs of 2019. So he is continuing this research with support from the Bill and Melinda Gates Foundation. So Professor Tahmid Ahmed, who is director of the International Center for Diarrheal Disease and Research, Bangladesh, a very prestigious center, as all of you know, is going to be talking to us this morning on an abnormal gut microbiota assembly is responsible for environmental enteropathy and stunting in children. I hand over to Dr. Tahmid Ahmed, please.
Thank you very much, Dr. Datta. A very good morning to all of you. First of all, I would like to thank Professor Nirmal Ganguly, whom I can see sitting in the audience, for the very kind invitation. Dr. Ganguly was not only a member of the Board of Trustees of ICDDRB, where I work, he is also a mentor to me, as well as many of me in Bangladesh. So thank you very much, sir. Thank you, Dr. Nirja Hajela. I can't see Dr. Balakrish Nair. And again, I must thank him because he has been my very close colleague at ICDDRB, who has spent many years trying to, improve what we have, a very renowned facility under the Laboratory Services, Science and Services Division. The kind of interaction, the kind of personality, which many of you will agree with me that Dr. Balakrish Nair has, we cherish still today, many years after he has left ICDDRB. So thank you very much for the very kind invitation. And with that, let me try to put up the slides.
Excuse me, I have come back to the.
Oh, okay.
There was some error in the link.
Okay. So can you see my slides, Dr.
Yes, we can see your slides and we can hear you very well, Dr. Ahmed.
Okay. So this is what I am going to talk to you about, environmental enteropathy, stunting in children, and the state of the gut microbiota assembly. And you can see at the top of this opening slide, I have a picture of the gut mucosa, and which is so very important, and I will talk about that. At the bottom, you can see a cartoon depicting the, different, childhood populations in low and middle income countries. And if you focus on stunting, which is characterized by linear growth faltering, as I speak to you, we have little less than 150 million children under the age of 5 years who are suffering from stunting. And it's not just the physical stature. These are children who have an increased risk of death, and those who survive, they suffer from, impaired mental development and poor intellectual, capabilities. So that's a very serious problem for low and middle income countries. This is a child whom we have been following in one of the slums in Dhaka city. And you can see over a number of years, the physical stature of the child has virtually remained the same. And this is what, stunting epitomizes in children living in Bangladesh, India, Pakistan, Nepal, Afghanistan, and many countries in sub-Saharan Africa. That is why, if you look at the SDGs, one of the very important targets of SDG Goal 2 is reduction in the number of children with stunting. And by 2025, we need to cut down the number of stunted children by at least 40%. Now, this can be a tall order for many countries because if you look at the track record, many of the countries unfortunately will not be able to, reduce the number of children by 40%. So back in the 70s and 80s, there was some interest in why children, they don't grow tall. And from a very small study done in the Gambia, it was found that, more than 40% of the linear growth faltering in children under the age of 2 years can be explained by what we call today the small gut enteropathy. Many years ago, we used to call it tropical enteropathy because people used to come to Bangladesh, India, Pakistan, and they would then suffer from, repeated attacks of diarrhea, growth faltering. They'd go back to their countries. After 3-4 months, they would recover. Then it was realized that it's not just tropical countries. This condition can be found in many other countries outside the tropical zone. So they started calling it environmental enteropathy because the reason is in the environment, as I will explain it to you. And very recently, there has been a new name. So it's a change of names every, 5-6 years or so. So now we call it environmental enteric dysfunction. And today, I am now going to tell you what this means and what has that got to do with the small intestinal microbiota. As you can see on this slide, we have subacute inflammation of the small gut with abnormal small intestinal biopsy findings. The problem is that you cannot do biopsy in all these children. So you have to rely on some other robust bedside markers by which you can say, yes, this is a child with stunting who has environmental enteropathy. This is a child who does not have environmental enteropathy. So these are the two pictures of a child in whom we did the endoscopy recently. And you can see on the left side of the panel, a healthy mucous membrane. And you can see the very slender, delicate finger-like projections, which is actually the villi. And it is through these villi that a child absorbs the nutrients that go into the system. But when the small intestine is colonized by bacteria, repeated colonization, when you have a toddler who moves around, touches the floor, and the floor is littered with pathogenic bacteria, places his hands in the mouth, and the bacteria go into the small intestine, with repeated exposure, there is colonization. And on the right side, you can see what happens. Total distortion and disruption of the villus architecture. When this happens, there is also chronic inflammation and ultimately malabsorption and malnutrition. These are the hallmarks of environmental enteropathy or environmental enteric dysfunction. We also have seen that, when mothers prepare the complementary food, be it in the slums of cities or in the villages, they prepare perhaps once in the morning and they keep the food. And in course of time, the food becomes contaminated. And we have been able to document that this complementary food, which is required for the growth of children, this is contaminated with pathogens such as E. coli. We also found that this is attributable to faulty food preparing practices. And ultimately, this results in higher rates of diarrhea and malnutrition. Sanitation, water sanitation and hygiene, that's a major issue. Fortunately, a country like Bangladesh has drastically reduced open rates of open defecation, so much so that the rate is now less than 1%. But if you look at this pie chart, you will see that even today we are talking about, in a city like Dhaka or in other major cities of the country, we are talking about 40 to 50% of latrines that are not optimal. So this means that we still have problems of sanitation. So coupled with the faulty preparation of complementary food, the absence or the suboptimal hygiene, and then the poor sanitation, this all contributes to an environmental condition that is conducive to what we call the environmental enteric dysfunction today. So what is known today? So we have so far relied on non-validated fecal or plasma biomarkers. And again, we don't have any biomarker that is considered as a gold standard. The upper GI endoscopy is something which is rarely performed because it is difficult in young children. There are ethical issues and other things. And then you can see that, we know that the perturbation of the gut microbiota contributes to undernutrition. We have shown that several years ago in case of severe acute malnutrition. But again, very recently, data suggests a potential role for bacteria in the upper gastrointestinal tract. With this, we started the Bangladesh Environmental Enteric Dysfunction Study, the BEAD study, where we wanted to validate currently available non-invasive biomarker candidates. We wanted to study the role of EED in malnutrition, particularly stunting, the biology of EED to identify common biological pathways for potential interventions, which to us is very important because till now, there is no single intervention that can take care of childhood stunting. We wanted to validate a system for histological scoring of EED, and finally to test the effectiveness of a nutritional intervention in taking care of children with stunting, with and without EED. So here you can see children 12 to 18 months old living in slum settlements in Dhaka city. And we had two groups, children with stunting, which is characterized by length for age z-score less than -2. Another group of children with milder degree of stunting, a LAZ of -1 to -2. And then we gave them a nutrition intervention. What is that? On-site feeding with 1 egg and 150 ml of milk, along with multiple micronutrients sprinkles. So this was provided on-site 6 days a week for 3 months. Okay. And at the end of 3 months, the children were again evaluated whether they had improved or not. If they had improved, graduation. If they have not improved, then we screen these children again for potential secondary causes of malnutrition. These include, draining ears, otitis media, tuberculosis, persistent infections such as urinary tract infections, so on and so forth. If they had no secondary causes of malnutrition, then we thought that they would be eligible for upper GI endoscopy. So with consent from the parents, we went for esophagogastroduodenoscopy. We took the biopsy, we took the duodenal aspirates, and we also had the histopathology. I don't want to bother you with this very complex slide. Just wanted to tell you to do an endoscopy on a young child, you had to go through all these steps. And you can see that starting from the routine work, we had this study on gut microbiota, proteomics, metabolomics, flow cytometry, transcriptomics, and the usual immunohistochemistry. Just to give you a glimpse of the methods, and here you can see right in the middle of the slum settlement, some of our colleagues from the study, the field staff talking with the mothers. And this is what we established in the middle of this slum, a feeding center where mothers could bring the children, spend some time, give the child the milk and the egg, which you can see here. And this is the micronutrient powder sachet, the mother feeding the child. And then comes the endoscopy. And this was done in a very good hospital because this was a study and we had to ensure that the very best was available for these children. As I said that, this study was done from July 2016 to July 2018. We enrolled 525 children, and of them, 110 children, they were eligible for the biopsy, for the endoscopy and biopsy. And you can see the different numbers. And finally, we had 80 children on whom we could also collect the duodenal aspirates because we were interested to look at the bacterial population, whether that's present in the duodenum of these children. So what we did, we looked at the duodenal proteins, we looked at the levels of plasma proteins, we looked at the small intestinal microbiota, the histopathology severity score, and of course, the length for age Z-score. Additionally, working with Professor Jeffrey Gordon, we had a series of translational studies in a mouse model, and this was to study the causal relationship by transferring the enteropathy to germ-free mouse colonized with cultured duodenal strains that we obtained from the children with EED. I am going to talk about it very soon. So this is what we did in terms of, grading the histopathology of children with stunting and EED. We looked at the infiltration of inflammatory cells in the lamina propria. We looked for atrophy of small intestinal villi, and then hyperplasia or elongation of crypts, which is again a hallmark. When you have a problem at the mucosa, the crypts, they go into hyperplasia or elongation. But, very surprisingly, we did not find any correlation between the histopathological score and the length for age Z-score. And again, reinforcing our belief that perhaps it is a problem with the small gut microbiota that is causing the problem of environmental enteropathy. So we also looked at the plasma proteins, and we used a state-of-the-art technique called SomaScan or SomaLogic to quantify several thousand proteins, present in the blood as well as the duodenal fluid. And we compared the plasma proteomes of children with EED with children without EED who are apparently healthy. And you can see that by doing this, we analyzed more than 4,000 plasma proteins and more than 2,600 duodenal proteins. Okay. So in the study of the gut microbiota, what we did, we used 16S ribosomal DNA sequencing. So we identified the ASVs, which is the amplicon sequence variants, by which you take care of the artifacts that are introduced as a result of the sequencing. And based on the ASVs, we tried to classify groups of the identified members of the gut microbiota. So this is what we did, after we did the clinical study in Dhaka slum settlements. We took the gut microbiota that we identified and isolated from the duodenum of children suffering from EED in Dhaka. Okay, so that was done in the laboratory of Washington University, St. Louis, and this is what we call the Notobiotic laboratory. Okay. So these mice, they were gavaged with the bacterial strains, okay? They were also given a diet that is typically consumed by 18-month-old children living in the slum settlements in Mirpur, trying to make the condition as close as possible to what we see in real life situation in the slum settlements in Mirpur. As a control, we also took, mice that were reared in the notobiotic laboratory, but who were given a transplant of cecal microbiota of a conventionally raised age-matched mouse. So you can see in the panel below the scheme that we used for this, translational experiments. So we used CoproSeq, community profiling by sequencing, duodenal RNA-seq flow cytometry. We also tried to look at how the bacteria actually are translocated from the gut to the bloodstream to the spleen. And this was done by, again, by full-length 16S ribosomal DNA amplicons. And again, we did a lot of statistical, mirroring to analyze the data, and that included from maneuvers such as the independent components analysis to the usual, correlations, and then the singular value decomposition to look at the relationship between the duodenal proteome and the absolute levels of duodenal ASVs. Okay. So this slide shows you the difference between the children who did not benefit from nutritional intervention and those who also did not benefit from nutritional intervention and on whom we could do the biopsy and also obtain the duodenal samples. As you can see, the age, the nutritional status, everything is more or less similar. If you look at the histopathological score, again, there is no difference, as well as no difference in the median fecal biomarkers, alpha-1 antitrypsin, myeloperoxidase, neopterin. So we then characterize the composition of the duodenal microbiota in these 36 children with EED who had an available duodenal aspirate. So there was shotgun sequencing and finally the V416S ribosomal DNA that I talked about. As a result of that, this is what we revealed. A core group of 14 bacterial taxa were identified in at least 80% of the duodenal aspirates obtained from children with EED. So this is something for the first time, and this is unique. The duodenum of children is usually sterile, but from children with stunting and EED, we could isolate a number of bacterial taxa. So these are the 14 core duodenal bacterial taxa. You can see starting from Streptococcus species Gamella, and then Haemophilus, Rothia, mucilaginusa, Veillonella. These two are very important, and I will tell you why. And on the left side, you can see again the methods that we use to identify these members of the duodenal microbiota. We then looked at the relationship between the plasma proteins and the length for age of the children. What we found was a group of 5 plasma proteins that were very strongly correlated with linear growth faltering. There were 4 that were positively correlated with length for age, meaning if you have a higher value of this, the stature, the nutritional status is going to be better. And this includes IGF-1, IGF acid labile subunit, IGF binding protein 3, and then we have the procollagen C, which facilitates bone formation. On the other hand, IGF binding protein 2 that inhibits functions of IGF-1, and that had a negative correlation with length for age Z-score. In this slide, you can see the relationship between the members of the 14 core duodenal bacterial taxa as well as the LAZ. So in C, you can see the better the LAZ, the lower is the total bacterial load. In case of Veillonella, again, the lower the length for age at the time of endoscopy, the higher was the yield of Veillonella from the duodenum of these children, implying that these bacteria are in some way associated with poor length for age of these children. We then went on to look at the abundance of these taxa and how they are correlated with the mucosal inflammation. Okay. And this was done by the using a maneuver called singular value decomposition. Here in this part of this slide, you can see the correlation. At the top, the different members of the 14, taxa, namely Veillonella, Streptococcus, Rathia, Corynebacterium. And then on the right side, you can see the names of the different, proteins. For example, the cathelicidin antimicrobial peptide, and then the lipocalin 2. And you can see the colors, which means the deeper the color, the more important, more significant is the correlation. So what we found is that these 14 taxa are correlated with the proteins that are pro-inflammatory in nature. So in stunting, we had this, duodenal proteins, which, have an impact on inflammation. And the abundance of these proteins was positively correlated with these bacterial taxa, meaning this bacterial taxa had something to do with the inflammation that is found in environmental enteropathy. We also tried to look at the, how abundant these members of the taxa are in children with enteropathy and children without, children who are healthy. And this is when we looked at the fecal samples of these children, because of course, we could not do upper GI endoscopy on children who are otherwise normal. And what we found was that, again, important members of those that were found in the duodenum that were overly represented in the fecal samples of children with enteropathy, the fecal samples. So we then went on to ask the question whether this is an association or a cause. For this, we had to go to the Notobiotic laboratory. And again, to give you an idea, I think I told you about this sequence that we did with the transplantation of the gut microbiota, the duodenal microbiota, transplanting into mice that were born and reared in the Notobiotic laboratory. At the top, you can see a photomicrograph of the intestinal mucosa of the mouse that received the transplant of the gut microbiota from children in Dhaka who suffered from EED. And you can see the gross distortion as well as the severe degree of inflammatory exudate, the high numbers of inflammatory cells, in the mucosa. At the bottom, on the right side, you can again see the slender villi, and on the left side, you can see the somewhat distorted, the abnormal, duodenal villi in mouse with EED culture collection. So again, pointing out the fact that transplantation of the gut microbiota from children with EED that induced enteropathy in notobiotic mice in the laboratory. So to summarize, ladies and gentlemen, what we found was that more than 4,000 plasma proteins, as well as more than 2,600 duodenal proteins in children with histopathologic evidence of environmental enteric dysfunction. More importantly, we could also identify 14 duodenal bacterial taxa that were present in the duodenum of these children with EED. And these taxa actually correlated with duodenal proteins, which are involved in immunoinflammatory responses in these children. And we also noted that these 14 duodenal taxa were distinct in fecal samples from EED when we compared them with healthy children. So the question is, what is the solution? We have found that yes, there is an abnormal gut microbiota that is responsible for this very important problem that is perhaps the most common problem affecting children in the world. Certainly, awareness about WASH among people, among policymakers, development partners, population control, I cannot overemphasize for a country like Bangladesh, but we need to go beyond the usual indicators of WASH. We need to review our work plan on WASH and ask the question, are we really doing enough? And then finally, what we have already started working with Dr. Gordon, develop therapeutic targets that we can use for modulating the abnormal gut microbiota that is responsible for enteropathy in these children. So what I have said has been published last year in June as an original article in the New England Journal of Medicine. And again, this was done in collaboration with Washington University in St. Louis and was funded by the Bill and Melinda Gates Foundation. This is a photograph of the two teams, and you can see Dr. Gordon here and the entire team of ICDDRB which is contributing to this study as well as other studies on childhood undernutrition. Thank you very much again for giving me this excellent opportunity to share our experience with you. Thank you.
May I ask Dr. GB Nair to moderate this discussion, if it's okay with you, sir?
You are muted.
Dr. Nair, you have to unmute yourself.
Okay. Thank you very much, Dr. Tamid, for this really spectacular talk. I like the way you put the whole thing across of this environmental enteropathy. The talk is open for questions.
So I'll read out one of the questions that has come in the chat,
Okay.
Dr. Nair.
Yeah.
The question is, can Veillonella species be associated with malnutrition? And does this bacteria have a reverse role in the colon?
Okay, can I answer now, Dr. Datta?
Yes, sir, please go ahead,
okay.
sir.
Yes, what we found was that among other members of the gut microbiota, Veillonella seems to be very important in causing enteropathy in these children. We also found, particularly in the mouse model, that the colon, the small gut, was relatively unaffected. So we think that enteropathy is still a small gut problem.
There is another question for you. The villi of mice with transplanted EED microbiota perhaps seem to exhibit neutrophil infiltration. Is this observation correct? Or is there some other explanation for the observed histopathology?
The observation is correct. And neutrophilic infiltration, as well as infiltration of other inflammatory cells, that happens to be a hallmark of enteropathy. And this is why, chronic inflammation sets in, producing, malabsorption and ultimately malnutrition.
Tamid, I just have a question. What is the role of enteric parasites
In
diarrhea and other
Okay,
infections?
so yes, in diarrhea, the picture is still not very clear, but certainly if a child has overwhelming infestation with a parasite, then there will be diarrhea. There will be nutrient sharing, and there will be malnutrition. But again, when you look at the new studies, the results of new studies, one particular study relates to the effect of giardiasis. Okay, so it seems that the presence of giardia is not associated with malnutrition. But again, as I say, that there are mixed results. Some studies are saying that yes, these have an important role to play in malnutrition. Some they don't know. But I can also tell you that, several years ago, there was a classic paper published in Lancet, and this was a study done in Uganda. They had the school health days, and on these school health days, community study program, in fact, they gave anti-helminthic treatment, and they found that this anti-helminthic treatment had a very good impact on child growth. Unfortunately, this could not be replicated in some other studies. So this is where we stand today, Dr. Nair.
I think
Thank you.
Professor Ganguly, I think wishes to make a
You,
comment from the floor.
And firstly, Dr. Tamid, we are most grateful that you could come. And this symposium was Tamid Day because yesterday Dr. Gordon presented your data.
Okay.
and today you presented a fresh data, and it is very interesting. So I want to bring it out that not even in children, Dr. Gorbach, who worked in the tropical school in India, published that upper gut in Indians have very big abundant flora which is not found in the western gut.
Right.
And he said that this is related to so-called tropical sprue. The
Right.
definition has changed. It might be adult enteropathy,
Right.
so-called tropical sprue in our Indian subcontinent. What I want to bring out that yesterday when there was an intervention study in notobiotic mice with healthy microbiota, and then you did about in 15 children in Mirpur, and it showed and this diet was given a name, and this was very nice and uh, laid out a hope that you could perhaps change these. And actually, when I was in the Gates Foundation Grand Challenge, we funded the microbiota study, and Dr. Gordon's lab was funded for that. And so I feel that, and I asked him and he couldn't answer perhaps. You have also done a lot of work on the ready-to-use food, which I asked one day that, is it khichdi? And you said, no, it is halwa because it is sweet. It has got sugar also. So I said that, could this be, since this has been done in a very large number of Bangladesh children, and also Indian children are benefiting out of this. So could we add a microbiota mix in this food? Because there could be, if there are multiple complementary food, it will be very, very difficult for the population to understand and administer this. So are you planning a larger study by creating something which will hope, or which will, and these biomarkers, will they also talk about saving the brain, that is the brain growth, apart from the bone growth and other linear growth parameters?
Right.
Thank you.
Thank you so much, sir. Um, as usual, you have summarized everything for us. And there are 4 things that come out from your questions. The first is, sir, you mentioned about adult enteropathy. You are absolutely correct. After childhood enteropathy, we are now made to believe that there is also adult enteropathy. So we have now planned to do the work in adults, particularly in women. And why women? Because in a country like Bangladesh, sir, low birth weight is very important problem. And this stems from the, low rates of weight gain during the 3rd trimester of pregnancy. And we think that, again, there might be a role for this gut microbiota in this, poor rates of weight gain. So, sir, we are planning to do a study on adults, particularly women. You mentioned about this MDCF, microbiota directed complementary food. Initially, we did a small uh, pre-proof of concept trial that was published in Science. And following that, we did a larger clinical trial that had clinical endpoints, meaning, weight for length, weight for age, anthropometric endpoints. And that we have completed. And I am very happy to let you know, sir, that the results of this clinical trial have been accepted for publication again in New England Journal of Medicine, and we hope to see the paper published early next month. And following that, sir, I completely agree with you that we need to go for larger trials where we can simply give, pouches of this local food ingredients, which the mother can mix at home and give it to the child. So we are working on this for trials, not only in Bangladesh, but also in Africa. And also we are discussing with colleagues in India if we can do such trials there. Certainly, we are looking at how these local food ingredients can modulate the gut microbiota, not just on its impact on promoting long bone growth, but also in improving the brain development of these children.
Dr. Nair, there are a couple of more small questions. Do you want me to read them out from the chat box? One of them is there any data with supplementation by fermented milk products in supplemental intervention studies? And the second question by Dr. Ramakrishna is that are the abnormal bacteria the primary cause of enteropathy, or are they both secondary to some other abnormality in physiology? So if you can very briefly answer them, because we're running a little short of time.
Yeah. Oh, thank you. Yes, fermented food that has been used for ages. And personally, I am a believer in fermented food, but I think we need more studies to document the impact of fermented food. And I think there have been trials done looking at the impact of yogurt, but there are also other studies that are ongoing looking at the impact on growth. Whether the gut microbiota, whether this is the primary cause, as asked by Dr. Ramakrishna, yes, the translational studies do prove that yes, it is the abnormal gut microbiota that is responsible for the enteropathy. This is exactly why we went for these translational studies.
I have a question, if you allow me. Can I go?
Yes, please.
Yeah. I have two questions. At the outset, my appreciation. It was very good presentation.
Thank you.
We know that initial 1,000 days of a child after conception is most important for his or her nutrition. Can we correlate nutrition of initial 1,000 days directly with the gut microbiota? Number one. Number two, as you said, that Millennium Development Goal, in addition to stunting, wasting, and low birth weight, nowadays obesity, especially clinical obesity, is most important and surpassing these three malnutrition conditions. So could you find, or can we think, a direct correlation between gut microbiota, ED, with obesity in the same pattern as we talked about stunting and low birth weight? So these two queries for you.
Yeah. So very important questions, sir, that you have raised. In fact, the studies on gut microbiota were actually prompted by small studies done on the role of gut microbiota in overweight and obesity in the United States. And those were spearheaded by Dr. Gordon, as well as a couple of other scientists who looked at the relationship between two taxa of bacteria. One is called Firmicutes and the other is the Bacteroidetes. And it is the imbalance in the abundance of these two bacteria that, causes more harvesting of nutrients when a person, takes the food, and that leads to overweight and obesity. So this is what we know today about the role of gut microbiota and overweight obesity. So exactly the opposite may happen in case of undernutrition.
So, and another was initial 1000 days of a child, its importance for the nutrition after conception.
Absolutely important. Absolutely important. And as I said, that in order to understand that, we need to first look at what is happening to the gut microbiota during pregnancy. This is our next plan. And I think maternal nutrition is the next nutrition frontier. Very important, as you have rightly said. If we don't take care of the first 1000 days, we will not be able to cut down the rates of childhood malnutrition.
Thank you very much. Thank you.
Thank you.
Dr. Datta, will you continue?
Yes.
It's easier.
Thank you, Dr. Nair. Thank you very much. Since we're running short of time, I'll straight away go on to introducing the next speaker. It's my pleasure to introduce Dr. Satoshi Hachimura. He's Associate Professor in the Research Center for Food Safety, Graduate School of Agricultural and Life Sciences, the University of Tokyo in Japan. Dr. Hachimura is also the Secretary Board Member of the Japanese Association for Food Immunology and Administrative Board Member of the Intestinal Microbiology Society. He is the recipient of the Food Immunology Award for Basic Research in 2009. He has numerous publications in peer-reviewed journals, and his field of specialization is food immunology. So this morning, he will be speaking to us about lactic acid bacteria introducing infection via the intestinal immune system. So I hand over to Professor Hachimura, and I welcome him to deliver his talk.
Thank you very much for your kind introduction, and I'll try to share my slides.
Dr. Hachimura, we can see your slides. If you can go onto the full screen mode, we can see the full screen slide and we can hear you very well. Please go ahead.
Okay, thank you very much for providing the opportunity to talk in this beautiful symposium. I am Satoshi Hachimura from the Research Center for Food Safety, Graduate School of Agricultural Life Sciences and University of Tokyo. Now, today I would like to talk about the lactic acid bacteria in reducing infection via the intestinal immune system. So yesterday, as Professor Pat also talked about, although the intestine, I would like to point out that is an organ for absorption of nutrients. It's also a large immune organ. The intestine is well inside out and connected to the outside world. Pathogens and commensals, and we eat every day to consume much food components. So you may agree that many, they say more than half of the immune cells are located in the intestine. So this slide shows you the intestine as an image of immunological point of view. These are the villi, of course, for absorbing nutrients, but the epithelial cells, which line a monolayer on this villi, also have immune functions. And like in the small intestine, we have lymph node-like structures called Peyer's patches. And inside the villi called the lamina propria, although not shown in this cartoon, many cells are present. So this electron microscope picture of the Peyer patches, you can see the leaf-like villi, and the Peyer patch is hidden right here to uptake antigens. And these are the M cells, I will talk about them later, which can uptake foreign substances. And you can see that these structures are full of immune cells. Now this is a picture of the villi. The green part is epithelial cells, and the red cells are those staining for the IgA antibody. And you can see that so much IgA antibody producing cells are present, and other immune cells are also present. And so there are many, many cells in this lamina propria region of the villi too. So you can say that the intestine is a large immune organ. Now, this theme of the symposium is the role of gut microbiota and probiotics in reducing viral infections. So in a state of impaired immunity, like you're too young or too aged or under stress, we have a higher risk of infection. And what kind of infection? I've been using this slide for many years and saying that pathogenic E. coli or virus like influenza and rotavirus and norovirus. I, of course, did not estimate, and I'm not happy that we might have to add coronavirus to this list. But we have been trying to find ways by immunomodulation by food, in particular lactic acid bacteria, to reduce the risk of such infections. So first of all, I would like to talk about IgA antibody. So there are many isotypes of the antibodies, and in the blood or in the lymph node cells in peripheral, IgG is the dominant isotype. But in the intestine, IgA would be. The IgA is a dimer and has so high neutralizing activity, and it has a role in host defense to pathogens and toxins. Of course, you know right now that also control the commensal bacteria. And it can also prevent allergy by preventing the uptake of large molecules. So B cells in the pair patches become IgA producing cells, and they move to the lamina propria region to secrete IgA and distribute this IgA, and these IgA are transported across the epithelium to the lumen. So we are in, B cells will become IgA secreting cells, but many cytokines and other cells are involved in this process. So in the textbooks, they say the helper T cells are helping this, but concerning the intestinal IgA, rather recent studies have shown that the intestinal dendritic cells play a very important role in this process. And we and other studies, researchers have shown that factors produced by dendritic cells can enhance IgA production. We have reported about IL-6 and Mora et al. For retinoic acid, and Tezuka and Oteki's group have shown about nitrogen oxide, BAFF/APRIL. And so this slide is a very simple culture to show you the role of intestinal dendritic cells in IgA production. Is a culture with dendritic cells, T cells, B cells, and an antigen. And PDC is for peripatch disease, and spleen disease is a peripheral spleen disease. So you can see from the slide that only when peripatch disease are used, IgA is detected. The other isotypes can be detected by spleen disease, but only when peripatch disease are present, IgA is produced. So this can show you the important role of the intestinal disease in IgA production. So this dendritic cells can produce these factors to induce IgA. And so we were examining the possibility that lactic acid bacteria can enhance this response. So first, I'll talk about this Lactobacillus plantrum strain. The mice were fed the strain by a diet. And you can see that the IgA production in small intestinal tissues are increased by the feeding of this lactic acid bacteria. So we examined the possibility that if dendritic cells are involved, and so in this culture with periparasitic dendritic cells and B cells and lactic acid bacteria, you see that only when dendritic cells are present, the lab can enhance IgA production. So we next examine what kind of IgA-induced factors are expressed. And in cultures, simple cultures with dendritic cells and bacteria, you can see that although production of TGF-beta or BAFF are not changed, production of IL-6 is increased by this lactic acid bacteria. And finally, in our culture with dendritic cells, IgA, B cells, and lactic bacteria, we added neutralizing anti-IL-6 antibody. And you can see here that although control antibody has no effect, adding the anti-IL-6 antibody abolished the effect of this bacterial strain to enhance IgA. So you would ask me, does this really provide protection? So in this slide, the bacteria was fed again as a diet. And you can see here that it does provide infection to the influenza, mouse influenza, and the mice who fed this lactic acid bacteria survive. And interestingly, not only in the intestine, by feeding this bacteria, you can enhance the IgA production in the BAF, which is the lungs. So as Dr. Pata pointed out yesterday also, and there's a common immune system in the lungs and intestine. Although we have not been able to prove, it's difficult to prove, but we think that the IgA positive producing cells in the intestine may have moved to the lungs. So these are in our animal studies. I've shown you that the lactic acid bacteria could act on dendritic cells. In our case, it's IL-6, but in other cases, like TGF-beta has been reported also, to enhance IgA production, which leads to prevention of infection. Now for next study, we went on to examine a different lactobacillus paracasase strain and on its effect on antigen-specific IgA production. So again, this strain was fed to the mice by diet, but the mice were orally immunized with a model antigen ovalbumin OVA and colartaxin as an adjuvant. So colartaxin enhanced the antibody production to OVA. So you can see in this slide that not only total IgA production in the intestine, but antigen-specific IgA, that's oval-specific IgA, in the intestinal tissues are enhanced by feeding this bacteria. So, and I was going to introduce these T follicular helper T cells, TFH cells, as a relatively newly investigated helper T cells member, but yesterday, Dr. Pillai showed us very, very interesting stuff about COVID-19 concerning the TFH cells. This T follicular helper cells are helper T cells that can move into the B cell region of B cell follicles of lymph nodes and in the intestine peripatesis. So we were considered the possibility that this lactic acid bacteria strain can be affecting this T cell subset. And so this slide shows you that feeding this lactic bacteria not only enhances the frequency of IgA positive B cells in the peripatches, but also the TFH cells that is identified by the CXCR5 positive and PD-1 high phenotype are also enhanced by feeding this bacteria. So I've shown you that this lactobacillus strain by feeding them, can enhance the induction of TFH cells in the peripatches. And we are doing some in vitro studies right now, but we think that in this case also, dendritic cells might be playing, acting, because dendritic cells present as the T cells to induce this TFH cells. Okay, and I've been talking about IgA so far, but another member, very important in antiviral infection, is the natural killer cells, NK cells. So NK cells are a long-known member of a rather newly identified family of innate lymphoid cells. They can kill or alter cells, maybe, like virus-infected cells. So there have been many reports to show that lactic acid bacteria or milk fermented with lactic acid bacteria could enhance NK activity. And I'm showing here about milk fermented by Lactobacillus casei shirota. And in this study, ingesting these fermented milk for 3 weeks, after ingesting, you see that the SCS fermented milk ingesting group have higher NK activity compared to the placebo group. And this figure shows you here that the change of NK activity by intake is higher, and those before the intake have lower NK cell activity. So it's effective in those with low NK activity, cell activity. In the low panel, this shows that in culture of peripheral blood mononuclear cells, PBMCs, when you add this strain to the culture, you can also enhance NK cell activity. And this shows you that this NK cell activity for each individual is correlated with the ability of the LCS strain to enhance IL-12 production. In this slide, it shows that the role of IL-12 in this enhancement of NK activity. And so in the culture of the PBMCs, when you add anti-IL-12 neutralizing antibody, you can abrogate the enhancement of interferon gamma or NK activity by adding this lactic acid bacteria strain. So these and other studies show that concerning the mechanism of NK enhancement by this casei strain is that the strains can act on macrophages, cells like macrophages, to induce the production of IL-12, which would activate, enhance NK activity. So there are many studies that show that this strain can have effect on infectious disease. One of these in middle-aged office workers, they are drinking this fermented milk of the LCS strain for 12 weeks. And you can see that concerning common cold, very significantly, the incidence is decreased. In case of influenza, well, it's not statistically significant, but due to the low incidence. But you can see that the percentage has lowered from 10.6 to 4.1. And this shows you in those subjects in that paper, NK activity and IgA and cortisol in the salvia. You can see that the NK activity at 6 weeks significantly increased in those drinking fermented milk, although IgA production is not changed and the levels of cortisol are decreased in those drinking the fermented milk. So I just told you about the common cold and influenza, but ingestion of lactobacteria or milk fermented or yogurt can have effect on other virus infections. And concerning this Lactobacillus casei shota, there are reports of effects on HIV or norovirus and HTLV1. And related maybe to these interesting effects of immune modulation, there are reports like marathon runners, the fermented milk could modulate the immune inflammatory responses. Or this study shows that fermented milk can prevent the physical symptoms in medical students under academic examination. There's medical stress, students are under stress of testing. And so such effects may be also related to the immunomodulation effects. So what you might ask me is NK enhancement by such strains like Casei shota, are they the intestinal immune system involved? Well, this may be a topic for future studies, but like this strain also uptaken by the peripatch and M cells I just told you in the first part of my study. And so they are taken up by the peripatches. So I would consider that in these cases too, the intestinal immune system may be involved in the effects of lactic acid bacteria. And I've been showing you what the possibilities in animal studies that lactic acid bacteria might directly act on cells like dendritic cells. But in yesterday's Dr. Oishi's presentation, I was very encouraged to find that in human studies too, the ingested probiotics are present very abundant in the ileum of human. But of course, the lactobacilli could maybe modulate the intestinal microbiota or metabolites, and this may be in the larger system be more important. But I think the cells the dendritic cells like macrophages to act on these cells are important, and they would affect then T cell, B cells, or NK cells. So this is about the end of the talk. And so I've been showing you that lactic acid bacteria may reduce the risk of infectious disease by like IgA, enhancement of IgA production, or enhancement of NK activity. And IgA production may be related to the effects on dendritic cells or TFH cells. And I hope, although I don't have any evidence, but these modulation may also have good effects on COVID-19. But in any case, lactic acid bacteria can modulate other immune responses also, like anti-allergic effects or anti-inflammation and anti-cancer. And I would presume that much of these activities can be through the intestinal immune system. So that is all. And I'd like to thank my collaborators, Kanshida and Masanano of the Yakuto Central Institute, and Kikuchi, Kunito Asari, Arai, and Iwabuchi, and my mentor, Professor Shuichi Kamioka. Thank you very much for your attention.
Thank you very much, very much, Dr. Hachimura. That was a brilliant presentation. And you very beautifully illustrated the important role of the dendritic cells in the Peyer's patches and how IL-6 modulates their response, their effect. And very nicely showed how the gut and the lung have some commonality in their immune system. And it was nice to hear that the interventions like Lactobacillus paracasei can reduce viral infections, and you again very nicely demonstrated the role of the T follicular helper cells and NK cells in acting through IL-12. And you summarized very well the complex interplay between lactic acid bacteria, the endogenous bacteria, and the various aspects of the immune system. We have a question for you in the chat box. It is that, and this was, I think, almost expected, that what are the pros and cons of using genetic engineered lactic acid bacteria as a prophylaxis against COVID-19?
Genetic modified, you mean?
Oh, well, any supplemental lactic acid bacteria as a prophylaxis against COVID-19? Any evidence?
Well, yes. Well, I was yesterday. Dr. Pot was talking about the good bugs and bad bugs, and I agree with him that although I'm not a physician, but inflammation is a very important, or the bad things in COVID-19, and so those cells which are those lactobacteria which can inhibit inflammation has also a possibility to have a good effect on COVID-19. I'm still optimistic about those NK cells and IgA, although not studied so far considered COVID-19. And yesterday, I have to admit that I did not know about the TFH cells defect in the COVID-19. And so I was very hoping that such effects of TFH inducing lactic acid bacteria may have a good role. And in Japan, I did not talk today, but there are food that containing not Lactobacillus, but Lactococcus strain that can activate the plasmacytoid dendritic cells. And these, of course, are very important antiviral immunity. And I think, although I also agree with Dr. Pot that it's best that we don't have any need, but I think that there's a very promising candidates to combat COVID-19 and lactic acid bacteria.
Okay. Professor Ganguly, I think you want to ask a question.
Dr. Hachimura, very nice presentation for food immunology. And when I was in PGI, I tried to infect animals, mice and monkeys with H. pylori, Helicobacter pylori. Unless we remove the Lactobacillus resident flora, we couldn't infect. This was an absolute necessity to remove the lactobacillus from the resident lactobacilli. So they play a very important role. I totally agree with you. What was new to me that I thought that the immune switches and immune responses, whether it is IgG or IgA or IgG subtypes, were modulated by interleukin 12. Interleukin 6, which is one of the inflammatory cytokine, modulates IgA. This was a little new to me. Could you give us the hypothesis through which it does, that how it does this function?
You're asking me about the role of IL-6 in IgA production, is that right? Yes, I know that IL-6 is often shown as a pro-inflammatory cytokine, but it also can enhance the. It's very well known that enhance the production of antibody from plasma cells, and when you do the study of dendritic cells, it has very efficient inducer of IgA. And my opinion is that those controls, IL-6 is important in control the immunity when it's under control. We need to provide IL-6 to the B cells. But of course, when inflammation, of course, many cells are going to produce IL-6 that usually do not, and that is not a good state. But I think that in controlled immunity, IL-6 have a role, important role in IgA production.
Dr. Hachimura, I have a question for you myself. You did mention about this gut-lung axis of IgA production, that when you provide lactic acid bacteria in the gut, you're also finding this in the lung. So did you also find it in other mucosal surfaces like salivary antibodies or in conjunctival tear? Once you feed lactobacilli, are you finding an immune response in other mucosal surfaces other than the lung?
Sorry, in my case, I only examined the lung because we were using the influenza infection model. But I think has been everybody's although the scientists are considering that lichen salvia is also regulated, and feeding some strains in human studies of lactobacillus can also enhance the IgA secretion in salvia. So I think, well, I don't have my proof by myself, but I think those are all connected.
Right. There is a question from the audience who's there in Delhi. Please go ahead.
Yeah, so nicely presented with different immune cells interacting with lactobacillus. So there is another immune cell, like mast cells, as you know, 2 to 3% on the mucosal surface of the intestine. And just I wonder what kind of interaction this bacterial population, lactobacillus or any other probiotic, towards inflammatory response or allergy response?
I'm sorry, I could not. Mast cells you're asking?
It's about the mast cells, interaction, Thank you. Mast cells.
Or in mast cells. In those strains, I told you I have not examined any interaction with allergy. But in the strain I told you about the TFH inducing activity. This strain also has anti-inflammatory activity. And so in the animal studies of influenza, it also can inhibit inflammation. So, well, I don't know the mechanism yet, but I think it's possible that both enhancing antibody responses and anti-inflammatory properties may coexist. But that's all I know about right now.
Dr. Hachimura, there are a few more small questions in the chat box, and if I can request you to answer those questions in writing in the chat box itself, that would be nice, because we do have to move on to the next lecture because we're running about 20 minutes late. So if you can answer those questions in the chat box, I would be really grateful to you. And I would like to thank you once again for a brilliant talk and for illuminating so many new issues. Thank you very much, Dr. Hachimura. With that, we
Thank you
will
for-
move on to the next exciting lecture. This is going to be delivered by Dr. Stephanie Jeanson. Dr. Stephanie Jeanson is the Science Activation Senior Manager at the Danone Nutricia Research Center in France. Dr. Stephanie Jeanson has a doctorate in pharmacy and a master's degree in toxicology. Initially, she had worked in clinical trials departments, and then she joined Danone Nutricia Research in 2008 to work on the safety of functional ingredients. Her role was to ensure the safety of added ingredients, including micronutrients, macronutrients, fibers, and strains. And she is currently working at the Dairy and Plant-Based Research Department to work on scientific communications around probiotics and gut microbiota. I welcome Dr. Jeanson, and I invite her to deliver her talk.
Thank you very much, Professor Dutta. Thanks for this nice introduction. So good morning, everyone. So I'm very pleased to be with you today, even if remotely. And I would like to thank the organizers for giving me the opportunity to present today our latest results. So as it has just been said, my name is Stephanie Jeanson. I work at Danone Nutricia Research in France in the R&I department. And I will talk about our latest results. And let me share my screen. It will be better. Should be okay.
Yes,
can you
we
confirm?
can see your screen and we can hear you very well. Can you just make it full
Perfect.
screen, please?
Yes.
Yeah, thank you. We can see your full screen. Please go ahead.
Thank you. So our latest results and my talk today is about the effect, a systematic review and a meta-analysis on the effect of a probiotic dairy drink containing the L-casein CNCMI 1518 and the yogurt strains on common infectious diseases. So this work was conducted in collaboration with Intertech Health Sciences and was published last November in Nutrients Journal. But first, some facts and figures about common infectious diseases or CID. What are we talking about? CIDs, we agree that this definition includes respiratory and gastrointestinal tract infections, such as the common cold, bronchitis, or gastroenteritis, for instance. These CIDs commonly occur in the general population, especially during winter, and are considered as a public health concern worldwide. In 2017, for instance, more than 17.5 billion of respiratory infections were reported worldwide, and more than 6 billion cases of infectious diarrhea. This disease, generally not severe, but they can lead to discomfort and economic losses due to missed day at work because you are sick or someone in your family is sick. And also it leads to some medical care, including antibiotics treatment and sometimes until the hospitalization. For all these reasons, prevention is key. Promoting good hygiene practices, promoting vaccination, improving living conditions and nutritional status are means of prevention. In particular, nutritional intervention trials have investigated the benefit of probiotic consumption in the management of common infectious diseases. Indeed, several systematic review and meta-analyses have been published over the last few years on the effect of probiotic consumption on CIDs prevention, treatment, or duration. Here, two examples on the prevention of respiratory tract infection in children specifically, or in the general population, which include children, adults, and seniors. In the first study, When pooling the result of 17 randomized control trials, the authors showed that probiotic consumption significantly decreases the number of subjects having at least one respiratory tract infection. The relative risk here of 0.89 with a p-value of 0.004. In the second study, so in the general population, the author concluded also of a benefit of probiotic consumption in the general population. So both studies really concluded that probiotic consumption may be beneficial in preventing respiratory tract infection. However, these two studies and this meta-analysis generally pool the effect of different probiotic strains from different families, mainly Lactobacilli and Bifidobacterium. Therefore, the question of a strain-specific effect may arise. In this context, one specific strain, the L-Casei CNCMI 1518, has been studied in clinical trials since 1998. In different studies conducted either in children, adult, or senior population, we have seen some effect on immune functions and immune parameters. Here, two examples of effects observed in children population and in an adult population. The first study is a randomized, double-blind, placebo-controlled trial conducted in children aged 3 to 6 years attending daycare centers or schools. For 3 months, they consumed twice a day a fermented dairy drink containing the L-Casei and the yogurt strains. We showed some promise in reducing the overall incidence of illnesses with the consumption of the dairy drink. Particularly, children who consumed the test product had a 24% lower incidence rate of gastrointestinal tract infection and an 18% lower incidence rate of upper respiratory tract infection, such as ear infections, sinusitis, pharyngitis, and laryngitis, compared to the control group. In the second publication, the study population was healthy adults working in 2 or 3 shift work patterns, including night work. Night work and shift work are associated with high stress levels, and we know that the risk of infection may be increased in people under stress. So this population was particularly selected because it was at a higher risk of having infections. So in this study, we tested again the effect of the probiotic dairy drink containing the L-Casei and the yogurt strain. The adults, the shift worker, consumed it twice a day for 3 months. And we also showed a significant reduction in the cumulative number of common infection diseases per class. So globally, generally, in both studies, we showed a reduction in the incidence of common infectious diseases. It was therefore hypothesized that the L-Casei CNCMI 1518 may reduce the risk of having CIDs. We conducted a systematic review and several meta-analyses to assess the effect of this fermented dairy drink containing the L-Casei and the yogurt strain on CID in the general population. Among the study inclusion criteria, we have considered randomized control studies on a general healthy population, including adults, senior, and children older than 2 years. We have considered studies whose test product was the fermented dairy drink with the L-Casei and the yogurt strains. And we have considered studies with outcomes related to CIDs. We ended up with 8 articles that included 9 clinical study results. The data quality of each study was checked according to the criteria of the National Institutes of Health. And globally, among our 9 studies, we had studies qualified as poor, fair, and good. And for different reasons that I will explain you just after, all study results could not have been pulled all together to perform a single meta-analysis. So, for our meta-analysis, we decided to focus on 3 outcomes: the CID incidence, the CID duration, and CID severity. As you can see here, with the incidence outcome, the metrics used to assess the incidence varied widely across the studies. If the 9 selected studies evaluated the incidence, it was assessed in many different ways. 4 studies assessed the relative risk, others looked at the mean number of CIDs per subject, or others assessed the incidence of specific disease or symptoms, such as diarrhea, vomiting, or abdominal pain. So finally, we conducted 3 meta-analyses to assess the CID incidence, one for each of the following metrics: the relative risk, the mean CID per subject, and subject with at least 1 CID. And for the other outcomes, since they were assessed only in 1 study, it was not possible to use them for meta-analysis and to combine the data. So because also of this variability of metrics, there was a scarcity of data by age group. So we had to combine the result of studies from different age groups. So you will see we combined studies as the result of studies of children, adults, and elderly. For the duration, the same challenge occurred. Here you can see for the duration outcome, we got 4 different metrics across our selected study. And 2 metrics were assessed in 3 studies, and these 2 metrics were thus considered later on for conducting the meta-analysis. These 2 metrics are the mean cumulative days of CIDs among subjects with CIDs and the mean days per CID episode among subjects with CIDs. And finally, but not shown here, for the severity outcome, we had the same issue. The variety of metrics was also important. Among our 9 studies, we ended up with 6 different metrics to assess the severity, but only one was common to 2 studies only, and the others were really specific to 1 study. So as a consequence, only 1 metric was considered for conducting a meta-analysis, and this metric is the cumulative number of CID categorized as severe among subjects with CIDs. So now I will present you the result of the different meta-analyses. So first, let's start with the incidence outcomes. As I said, due to the variety of metrics, we had to perform 3 meta-analyses on the 3 metrics used in more than 1 study. And we also combined the results among the different age groups. As regards to the first meta-analysis, to assess the relative risk, 4 study results were combined: 2 in children, 1 in adults, and 1 in seniors. Compared to the control, we showed that the consumption of the fermented dairy drink containing the LKCI, CNCMI 1518, and the yogurt strain twice a day resulted in a trend towards a reduced risk in cumulative CIDs. So here in this first graph. So a reduction in the relative risk but not statistically significant. For the second outcome, the second matrix, the mean number of CIDs per subject, we combined the result of two studies, one in elderly and the second one in adults. For this metric, the consumption of fermented dairy drink significantly reduced the mean number of CID per subject compared to the placebo, so with a p-value of 0.001. And finally, for the assessment of the odds of experiencing at least one CID, we combined the result of three studies, two conducted in elderly and one in adults. Again, in this case, the consumption of the fermented dairy drink significantly reduced the odds of experiencing at least one CID compared to the control in the study population, with an odds ratio of 0.81 and a p-value of 0.029. So as regards to the assessment of the CID duration, we conducted two meta-analyses on two different metrics. For the first one, the mean cumulative days of CID among subjects with CIDs, three studies were combined on elderly and children population. There was a decrease in the mean cumulative days of CID when consuming the fermented dairy drink, but this difference was not significant. So the results are shown here. For the second metric, the mean number of days per CID episodes among subjects with CID, we also combined the result of three studies in children, adults, and elderly. In this case, also, even if we saw a reduction in the mean duration of a CID episode, when consuming this product, this difference was not significant. On our third outcome assessed, the severity, the results are not shown here, but we did not show any differences in the severity of a CID when consuming the test product compared to the control. And as a reminder, this outcome was assessed by combining the result of two studies only, and the matrix assessed was the odds of experiencing common infection diseases categorized as severe among subjects with CIDs. As a conclusion, this is the first systematic review and meta-analysis conducted specifically on studies with the LKCI CNCMI 1518. The result showed no effect on the probiotic dairy drink on CID duration and severity. But on the contrary, we showed significant reduction in the odds of experiencing at least one CID and in the reduction in mean CIDs per subject. This result suggests that the fermented dairy drink containing the LKCI CNCMI 1518 and the yogurt strain may reduce CID incidence in the general LC population. By conducting this study, some questions arose regarding the metrics, the methods, and the quality appraisal. As shown in the presentation, there were plenty of metrics used for one single outcome. In our case, across our 9 studies, we identified 7 different metrics for the incidence, 4 different metrics for the duration, and 6 metrics for the severity. It was therefore not possible to pull all study results. That's why an adequate reporting and presentation of the results for the CID's outcome could facilitate in the future the data pooling for systematic review and meta-analysis. It also appeared that the methods used to assess the outcome were not always well described and complete in the publication. This is, however, essential in understanding whether the study was susceptible to potential confoundings. And for example, in one study conducted in children, we had the case of having only parent-reported outcomes, which is not sufficient. And in this case, a diagnosis by a health professional should be considered to avoid any confounding factors. And finally, additional quality criteria were applied to the NIH tool. A list of potential confounders relevant to clinical studies on immune functions were considered to be of importance. For instance, we included as additional criteria the vaccination status or the concomitant medication that may alter, respectively, either the incidence or the duration of a CID. So some guidance or checklist of additional study considerations may be of interest to conduct and assess further studies. I thank you very much for your attention. I conclude with a picture, a nice picture with a nice weather of our research center in Paris-Saclay. Thank you very much.
Thank you very much, Dr. Stephanie Johnson, for that first ever meta-analysis systematic review on this particular issue. And you've also highlighted the challenges that you faced in doing this meta-analysis because there was a diversity of outcomes and very few studies for individual outcomes. And hopefully in the future, we will have more studies in this better conducted randomized controlled trials, and an updated meta-analysis in the future may show even more light on this. I was just curious to know your figures of the meta-analysis did not show the absolute numbers in each arm and the event rate. So I was just wondering what was the cumulative sample size that you were looking at when you showed these outcomes? Like for each outcome, what was a gross figure? Was it in a few hundreds or?
Yes, yes. The study conducted in children included more than 600 children per study, and for adults it was from 200 up to 1,000 people per study. So yeah, globally, for each meta-analysis, there were several hundreds of people per studies and per meta-analysis.
Yeah, so probably we would get more definitive data once these numbers go into several thousands. And So there are a few questions that have come up in the chat box. So one of the first questions is the limitations of the meta-analysis are obvious. Why not focus on the susceptible group studies with objective markers of the common infectious diseases? I'm not sure what this question means, but probably asking whether we should focus on high-risk populations. Yeah.
Probably, yes, it's a good question. I think the question is also why not using other markers than probably CID incidence. We decided to focus specifically on the study conducted with the LKZI CNCM I-1518. And in these studies, we rarely have that immune marker investigating the effect in the population. So we have clinical markers, the number of disease, the incidence, the severity, the duration, but we did not have enough data to pool to assess other markers.
Yep.
I don't know if I correctly answered.
Yeah. The next question is a very nice presentation. It is suggested, please try fermented whey products with probiotic lactic acid bacteria to improve gut flora. So I think the question is, in a few intervention trials, should you be combining fermented whey products with
fiber? Yes, indeed, it can be a nice try.
Yep. The next question is, could you kindly elaborate on the reasons for grouping the same study in different combinations with other studies in your meta-analysis?
As I showed, we faced plenty of metrics assessing different outcome, and it was not possible to pull all these metrics were not assessed in all studies. So that's why we could not pull the 9 study results into one single meta-analysis, because we did not have one common metric assessed in the 9 studies. So that's why we did several meta-analyses, pulling what could be pulled together based on the data we had.
Here's a question. Does the dose of the probiotic remain the same for all the age groups to prevent common infectious diseases?
Exactly. The dose was always 2 products per day. So with a dose of L-casein around 10 to the 10 CFU per day.
If I understood correctly, you were also looking at a combination of L-paracasein with your 2 yogurt strains?
In fact, the product tested is always a fermented dairy drink containing the L-casein and also containing the yogurt strains.
So is it possible then to tease out the individual impact of each of these strains?
This is actually this is a good question that we cannot do for now, because in the study that I presented here, the control product was an acidified milk, and it was not a yogurt. So the benefit of that we see here are related to the full product, so the L-casein and the yogurt strain, even if we think that we have proof that the L-casein alone has benefit on the gut microbiota, on immune markers, on promoting NK cells, etc., etc.
Okay. Are there any questions from the audience in Delhi who are physically present? I don't see any hands raised. If there are any hands raised, could the camera move to whoever? Professor Ganguly.
How long when the ultimate effects were not on severity or duration, ultimate effect was in the incidence or in the prevention of the CIDs. So how long you will have to take these products? And how long was the duration of the study? Like I understood there were 600 or up to 1,000 sample size, but how long they had to take the product?
The studies were conducted for a period of the subject consumed the product for at least 12 weeks. So starting from 12 weeks, then we have a bit more, some studies with 13 weeks, 4 months, but the majority of the studies were conducted on a 12 weeks consumption period.
And this was a randomized placebo control.
Exactly, except one study which was open label, which was excluded from the analysis.
Thank you.
If there are no more questions, thank you very much, Dr. Stephanie Jeanson, for that wonderful talk. We really appreciate your presentation. And with that, we will move on to the next session of the day, a session that we've all been looking forward to. This is the Young Investigators presentation. We have 3 presenters who have already been adjudged by an expert panel, and they have been awarded their prizes. So there's a first, second, and a third prize. So the prize winners will be presenting today. The first speaker is going to be Mr. Shubhrajit Bhad. He earned his MTech from the Central University of Hyderabad and is currently employed as a researcher in TCS Research, Tata Consultancy Services. His research focus is in the area of biomarker discovery and host microbial interactions. And he is going to be presenting his award-winning talk. I now hand over to Mr. Subhrajit Bhar. Could you please share your presentation? And you'll have to stay within the time limit that's been allotted. And I believe you'll be having 6 slides that you'll be presenting. Over to you.
Thank you. Thank you for giving me this opportunity to present in this forum, as well as showcasing some of the works that we do in TCS Research. So I will be sharing my screen over here. Yeah, I guess I am visible.
Yes, you're visible. Please go ahead.
Slides are full screen, right?
Full screen, yeah, and you're audible. Please go ahead.
Okay. So, yeah. Good morning, friends. I am Subhrajit Bhar from TCS Research, as I have been introduced. And as we have heard about the various aspects of the microbiome, that it has associations with various diseases and disorders, and it has been leveraged for various probiotic supplements, etc., as well as it has been several times leveraged as a diagnostic marker. However, in this case, the markers were mostly taxonomic markers, which are dependent on various factors such as geography, ethnicity, or diet. And it would be nice if we have some kind of aspect of the microbiome that actually is independent of these factors. To speak about such an aspect, I would be speaking today on the possibility of sensory protein signatures in the gut microbiome as biomarkers to detect diseases in the early stages, mostly during the onset when they are mostly asymptomatic in nature. So what are the sensory proteins? They are a group of proteins within the cell, which actually helps the cell to receive various stimuli from the environment and enables the cell to acclimatize to that environment by generating appropriate responses to the stimuli. It is very important for bacteria because the bacterial environments are continuously changing and they need to acclimatize to that environment in order to propagate in that environment. So in our previous work, we had demonstrated as to how sensory protein signatures, we had found that these signatures could identify between the non-pathogenic and the pathogenic strains of Escherichia coli. We further had modeled these for model-based predictions based on SVM modeling, and we used 100-fold cross-validation on 80-20 split data of the various E. coli strains we studied. And we found that in the test data, we had found that it could separate the pathogenic E. coli from the non-pathogenic E. coli with a sensitivity of greater than 90% and a specificity of about 60%, while AUC remained around 80%. So it would be intriguing now that in Escherichia coli, we have the sensory protein signature actually senses the virulence of the Escherichia coli. It would be intriguing to see whether it can sense any change within the microenvironment and whether it can reflect towards the health status of the host in case of a gut microbiome. Now, why we are doing this? Because for an example, if there is a cancerous tissue and a healthy tissue, the microbiome near the cancerous tissue is expected to have a different kind of microenvironment than the healthy tissue due to the various chemical and physical changes, etc. And in this case, we have found that in sensory protein signatures, the sensory proteins near the cancerous tissue would be somewhat different than the healthy tissue. So what we wanted to see was if the sensory protein signatures could indicate towards the onset or the progression of a disease during the onset when the diseases are asymptomatic in nature, whether it could identify the disease from the healthy cases. And for this, we had two data sets. One was for colorectal cancer, where we had healthy adenoma and carcinoma samples. And the other was for diabetes, where we had samples for healthy prediabetes and diabetes. And we use this into the model-based prediction techniques to get our results. So in the method workflow, what we did was we first created a sensory protein database where the database was found or derived from the annotated sensory proteins that were actually derived from the sequence genomes. And we mapped it to the other proteins which were poorly annotated or unannotated in nature, and we found around 87,000 sensory proteins which we kept as a sensory protein database. We mapped the metagenomic reads from the samples of these data sets that we used against this sensory protein database for the sensory protein profiles. The sensory protein profiles for each of the species was calculated as in the cumulative aligned base length of the sensory protein genes within that particular species divided by the metagenomic sample size. And then we created a sensory protein based abundance matrix in order to like a kind of a modified and abundance table, which we use for random forest modeling based prediction. And we split the data into 90% train and 10% test, where the 90% training data was used for 100-fold cross validation and tested on the 10% test data. So we come to our results where we found that our sensory protein signature based methods had a EUC better than the than the methods which were reported earlier that were based on taxonomy, especially in the adenoma and prediabetes cases. They performed pretty well than the taxonomy based methods. And adenoma and prediabetes are very hard to diagnose with the current techniques. And in this case, our technique can be useful as a companion diagnostic tool in order to find this kind of onset of the disease of carcinoma or diabetes. Even in the case of detecting diabetes from control, it performed well. Our sensory protein signature based models performed well than the taxonomy based models. And though in case of carcinoma, our sensory protein based signature based models were not as useful as the taxonomy based models. However, carcinoma can be detected with the current diagnostic tools fairly well, and it is more interesting to note that we are able to find out the detection. We are able to detect the adenoma and
just a gentle reminder that we have to wind up.
Yeah. Yeah, as I was saying, that it is more important to know that adenoma and prediabetes can be diagnosed by this kind of method. So we are coming to an end of the talk, and I will just summarize what we found, that the sensory protein signatures can indeed reflect to the changes within the microenvironment and somewhere reflects towards the host health. And we also found that the AUC based on the sensory protein based signatures based modeling methods were better than those used for the taxonomy. And the significance can be that these can be used as biomarkers for detection of various asymptomatic diseases during the onset of the disease. The amplicon-based sequencing method can be applied if we can sequence the kinase domains of the various sensory protein sequences and use this to reduce the cost of sequencing. The non-invasive technique can be useful in detecting most of the onset of the diseases that use otherwise invasive techniques, like for colon cancer, we have colonoscopy, which is quite invasive. And the timely diagnosis, lastly, of these diseases actually reduces the treatment cost and also reduces the mortality. Thank you. I would be open for questions if any.
Thank you very much. I don't see any questions in the chat box, but if there are any questions from the floor, from the audience who are physically present, I'll be happy to take up those questions. We're running short of time, so maximum maybe 1 or 2 questions. Is there anyone who wishes to ask a question from the floor? Okay, so
Because-
can I ask you a question? So
Yeah,
did you try
sure.
to find out an optimal cutoff value which you can apply in a validation cohort?
As for now, we have kept in the validation cohort, like we have used several seeds for the test data, and we have kept the median value that we are like we are not taking the best performing model. We are taking the median of several seeds.
Right,
Here.
because if you're trying to use it as a screening tool in asymptomatic, you will have to have very high sensitivity.
Yeah.
Even if that is at the expense of a low specificity. Okay.
Yeah.
Since there are no more questions, thank you very much for a wonderful presentation.
Thank you.
And we will move on to the speaker who has won the second prize, and that is Dr. Mangesh Vasant Suryavanshi. Dr. Suryavanshi has a master's in microbiology and a PhD degree in biotechnology from the National Center for Cell Sciences, Pune, and he has recently been awarded the SERB NPDF fellowship on which he's working, and he has more than 36 publications to his credit. I invite Dr. Suryavanshi to make his presentation. Again, please remember to stick within the time limits and the number of slides. Please start sharing your screen.
Sir, thank you so much. So, am I audible or visible?
You are audible and your screen is full screen is visible. Please go ahead.
Thank you so much for the kind information, kind introduction. So, I am thankful to the organizers for giving me a chance to present my work here. So, I'm going to show some observations related to the skin, oral, and gut microbiota, which shares some phylotypes to the urinary microbiome, and its divergence may associated with the types of urinary kidney stones. So basically, I'm working with the urolithiasis and the urine microbiome. So this is the small observations I'm going to put forward to you. So, so coming to the point that we know, we from fact that we coming from the stone belt area where incidence of kidney stone diseases, kidney stones in the general population is more than 15%. So this is important to know about what is exactly the pathophysiology of the crystallization event that happens if that due to the bacteria itself. So we know the fact that the UTI, that is a urinary tract, have a certain kind of ecology. So we previously thought that we don't have the microbiome flora with the urinary tract, but eventually we accept that ecology inside the urine have its own importance. So apart from that, we have a basic questions from Indian population, that is what is exactly the urobiome, urogenital microbiome, that is a UMB from the Indian population. Second is that why I mean, how this is urinary tract microbiome exactly transmitted from whether it is transmitted from gut to the this UTI, urine tract. So another one is that if that is there, but how much a diversion of this urinary microbiome with respect to the stone diseases. So those are the basic questions I am dealing with right now. So at the present, whatever I am going to tell you about the observations that is related to the present study. So we work with the it's like a cross-sectional study. You can say it's a case control study. So 369 total population have been studied here. So my primary objective has to be go with the one gut urinary axis, where you can easily know that some kind of phylotypes have been associated, between the gut as the gut to the urine itself. So so come to the observation part. We did urine culture as well as urine analysis, and other part goes for the NGS analysis. So whatever the patients who have kidney stone, so we removed the kidney stones and identified with the FTR and X-ray crystallography. So we specifically found that around 40% of stone samples and 36% of urine samples of the KSD patients have the positive for the urine cultures. That is a culture results. So apart from that, healthy urines also 29% for positive for the urine cultures. So in total, 44 bacterial species identify throughout my this study. So we identify those 44 bacteria species firstly with the 16S and goes with the WHS activities. Parallel to that, we did the NGS for the 16S sampling on studies. We found that total 542 genera have been identified inside the inside that environment. So we call them as a urinary microbiome. So as I told you, this is a case control study. We found that those KSD patients have 183 genera which are exclusively present in the KSD patients. So come to the point that is it possible to highlight the part what is exactly the core urine microbiome of our Indian populations? We found that 32 genera 100% present into the healthy environment, healthy cohort, and 11 genera which are present in respect with the KSD study, KSD incidences. So our next phase is that how we can trace whether those the phylotypes have the shade between the gut and other body parts. So we did source tracker analysis for that. We used skin, oral, and gut data set for this analysis. We found that more than 0.6% and more than 9% of this skin, oral, and gut phylotypes shares to the disease environment, and 13.3, 0.2, and more than 10% of this flora have shared with the healthy group. So our next observation is that through the cultural activities, we found that disease specific flora, we found that. So due to the 16S identification, we come to know that there are certain kind of flora exclusively highly abundant with respect to the types of kidney stones. So we made a certain observations related to that and come to conclusions that there are some biomarker kind of activities, biomarker species have been present in the kidney stone environments. So our next point is that
just a gentle reminder that you will have to summarize.
Yeah. Okay. So come to the point that we after CC analysis, we find that there are certain kind of let's say, for example, chemical composition of kidney stones have the major impact of that divergence activity inside the urinary microbiome studies. So so we have a working hypothesis that the bacteria have the ability to generate a kidney stone inside the kidneys. So our first report has with the Calamia species. So we developed the evidence for the this species as a non-urease producer and comes from the environment and have the ability to produce the Stewart crystallization inside the body. So apart from that, I'm a part of American Urological Association Microbiome Consortia. So we have a very different kind of questions related to the kidney stone diseases. So starting with the how it is the kidney itself, and then to the how the treatment value offers to the control such kind of incidences. So with the we have a team, and we started working on standardization of protocols, starting from the standardization protocols to the urolithiasis study. We also developed some concrete observations based on meta-analysis studies. So those publications are is underway now. So I would like to thank for this study my mentors like Professor Rekha PD from Nagoya University, Professor Yogesh Savche, and Dr. Aaron Miller from the Cleveland Clinic. So thank you so much.
Yeah. Thank you, Dr. Suryavanshi. We do not have any questions from the chat box. If there happen to be any questions from the floor, we can take that up. Okay, if there are none, I have just one question for you. So in clinical practice, there are 3 ways of taking a urine sample. Either you do a suprapubic bladder tap ultrasound guided, or you do a clean catheterization of the bladder, or you take, a periurethral sample. That means the patient passes urine into a collector. So which of the 3 did you use? Because I was intrigued that your normal healthy controls, about 30% grew culturable bacteria. So are we sure that these are not contaminants?
Yeah, so we collected the urine sample, that is a midstream urine sample, sir.
Okay,
so yeah.
So up to 10 raised to the power 5 bacteria in midstream urine per ml in midstream urine sample is considered normal because of contamination. So I just wanted to point that out. Yeah. Okay. Thank you very much, Dr. Suryavanshi. We will now move on to the winner of the 3rd prize. That is Dr. Ankita Kumari. Dr. Ankita Kumari has just recently submitted her PhD thesis, which was based on the study of epigenetic modifications of intestinal cells in the presence of probiotic bacteria. And that's where she intends to pursue her career. So I welcome Dr. Ankita and I invite you to make your presentation. Please start sharing your screen.
Thank you so much for the kind information, sir. Very good afternoon to all. So my screen is visible?
Not yet, but I think it's going to come because it says it's Ankita has started screen sharing. Yeah, it's come up now. Can you just make it full screen?
Yes, sir, it is full screen.
Yeah, now it's full screen. There's a little bit of a lag between your slides and coming up here. Please go ahead.
Okay. Thank you so much for the this opportunity. Today I'm going to talk. The title of my today's talk is the probiotic lactobacillus fermentum mediates its immunoregulatory functions by targeting epigenomic modulations. So let's start with the introduction with this probiotic, that is lactobacillus fermentum MTCC 5898, earlier isolated from the feces of 9-month-old infant and characterized for. It was found that the milk fermented with this probiotic, it lead to the enhancement of the systemic immune responses as well as the antioxidative capacity in the mice. And further, in case of the gut barrier functions, it was also found to improve the gut barrier impaired gut barrier functions against the infection of the E. coli. And in case of cells, intestinal cells, and also its role was also found in case of immune signaling and was found to be safe for the human consumption. But for the before the human consumption, it is important to know whether probiotic lactobacillus fermentum regulate the immune modulatory activities through epigenetic modifications. These modifications are the addition or the removal of some of the chemical groups like methyl acetyl by the different group of the enzymes, which lead to the remodeling of the chromatin structure and which further affect the gene transcription. So we have selected the one of the modifier, that histone modifier, that is histone deacetylase 1, and which is involved in the removal of the acetylation. Removal of the acetylation from the histone proteins and lead to the reduction of the RNA expression. In present investigation, we have what happened?
Dr. Ankita, your slide is not moving.
guess there is some technical fault. My slides are not moving. I think I should stop sharing first.
Yes, we can see your slide.
Yes, sir. Okay, sir. So in present investigation, we have inhibited this histone modifier by using its specific inhibitor MS275, and this the acetylation, so that the acetylation was remained same and the RNA expression was higher. So this was the experimental plan for the investigation, which were and for this we have chosen the Caco2 cells, which are considered as the best intestinal model for the experiment. And then the 4 controls were taken. As the one was the medium control, where the cells were not treated with anything, but the another controls, including the treatment with inhibitor, probiotic, or the E. coli, it was chosen as a positive control. In case of experimental group, the cells were first treated with the inhibitor at 2.5 micromolar concentration for 18 hours, and which were concentration and the timing were already standardized. And then they treated with the probiotic and the E. coli. And the assessment of the gene expression of innate immune genes were done by using real-time PCR, and the acetylation pattern was studied by using chip assay. This is the 10⁹ CFU for the probiotic and the E. coli, and the timing of the 6 hours. They were already standardized, where the immune genes gave their higher expression, and the 10⁹ CFU was the safe dose. And for the E. coli, the cells were live at the 10⁹ CFU for ml, and it was found that during the treatment with the inhibitor, there was a significant increase in expression of the all immune genes, that is interleukin 6, 8, and the human beta defensin 2, as per hypothesis. But when the cells were treated with either alone LF with the post-treatment of this probiotic after the pre-treatment of inhibitor, it lead to the significant reduction in the expression as compared to the inhibitor treatment of all the genes. And but in case of inflammatory agent E. coli, it was found that opposite to the LF, there was it increased the E. coli increased the expression of these immune genes, and when combined with the inhibitor, it lead to there was synergistic increase was observed in case of interleukin 6 and human beta defensin 2, and the much higher expression was observed in case of E. coli infection. In case of interleukin 8 gene expression. To further know whether these changes, as in slides we have seen that this gene expression was due to the acetylation in presence of inhibitor. So to further check this, the involvement of H3 acetylation, we have by using chip assay, it was found that in presence of inhibitor, there was increase in acetylation and the promoter region of interleukin 6, 8, as well as the human beta defensin 2 gene. But again, as similar to the transcription studies, the lactobacillus fermentum, when the cells were post-treated with this probiotic, it further results into the reduction in the acetylation level on the promoter region of these genes. So it was directly linked that the gene transcription was due to the changes in the reduced gene expression was due to the reduced H3 acetylation. And similar to the gene expression studies, the E. coli was further contributed to the higher acetylation level, similar to this inhibitor, and further with the combination of inhibitor, it further increased the acetylation on the promoter region of these interleukin 8 and HBD2. So it was concluded that the fermentum and E. coli, they have contrasting effect on the histone acetylation of the immune gene. Further, the role of histone deacetylase was seen as the expression of this gene was increased in the presence of fermentum at 12 hours of treatment during temporal gene expression, but the E. coli reduced its expression at 12 hours up to the half as compared to the control. So it was concluded that LF it act as the activator of the histone deacetylase, which further result into the deacetylation of the histones and the result into the decrease in RNA expression. So it can be further.
A gentle
So
reminder,
this
you'll have to wind
probiotic
up
has
in a
therapeutic
minute.
potential that it can be used in the immune therapies of different inflammatory diseases like IBD, ulcerative colitis, colon cancer, with disrupted pattern or generally higher histone acetylation on these immune genes were observed. Thank you.
Thank you very much, Dr. Ankita. I think there's one question in the chat. Any reason to use the same value for E. coli and probiotic cells for the experimental study? Maybe you can answer that in the chat box itself directly to the person who's asked the question. Because I get a message from Dr. Ankita saying that she's not able to hear me. So can you please answer the question in the chat box itself, Dr. Ankita? Since we are running late by about 15-20 minutes, we will not be taking any more questions. I would like to thank Dr. Ankita for her excellent presentation, and to all the 3 award winners and the 3 plenary speakers earlier this morning. And with that, I would like to bring this session to a close. I would also like to thank my senior co-chairperson, Professor G.B. Nair, for his initial comments and his gracious presence. With that, I hand over back to the organizers for starting the panel discussion, which is the next session of the day. Thank you all very much.
Well, thank you so much, sir. So now we move on to the one of the most exciting parts of the symposium, which is the panel discussion. The theme of the panel discussion is gut microbiota, probiotics, and immune response, the link, which will help us understand and address some of the questions that often come to mind about the emerging science of intestinal microbiota and probiotics. So, ladies and gentlemen, we would now like to invite Professor Anurag Kurpad to moderate the panel. I would like to highlight Professor Anurag Kurpad is professor and at St. John's Medical College, Bangalore, India, and was the founding dean of St. John's Research Institute, Bangalore, India. He's the head of the only IAEA Collaborating Center on Nutrition, located at St. John. He's a fellow of the Royal College of Physicians, the Indian National Academy of Medical Sciences, and the International Union of Nutritional Sciences. He has published over 400 papers and is co-author of the Asian adaptation of Guyton's textbook of physiology and co-editor of the Asia Pacific Journal of Clinical Nutrition. So ladies and gentlemen, please put your hands together for Professor Anurag Kurpad. Requesting sir to kindly take the panel discussion forward and introduce the panelists. Thank you.
Yeah, thank you. Can you hear me? I hope you can. It's a pleasure for me to be here and to be coordinating this panel discussion. We have some very fine panelists here who can bring together all the things we've been hearing up to now about probiotics. And I had something that would introduce all of them, but I seem to have lost that. So what I will do, on the other hand, is to simply tell you their names. And I think you all know them anyway, and we are short of time. So it'll be good if we can, once we know who the panelists are, that we move straight into questions for them. So our panelists are Professor Shashikaran Boindala, who was a former director of the National Institute of Nutrition. He's a pathologist of great renown, and in his work in nutrition, he worked a lot on probiotics and in setting the regulations for probiotics in India. We also have Dr. B.S. Ramakrishna, who again is one of the most renowned gastroenterologists in India. He's very well known for his work on the gut microbiota and has published extensively in this area. He's presently at SRM Institute for Gastroenterology in Chennai. We also have Dr. Srivastav, and Dr. Srivastav is truly someone who is a very, very passionate scientist and promoter of milk. He has worked a lot in the area of milk in India and the nutritional qualities of milk, and has also worked on probiotics. He was, of course, the head of NDRI, and that tells you why he was, and that tells you his abilities. We also have Dr. Prajapati, who is at the Anand Agricultural University, and he too has done a great deal of work on bioactive peptides and probiotics. We also have Dr. Anita Jatana, who is the chief dietitian at the Apollo hospitals, who has a lot of experience on the ground practical uses of probiotics. So let me start immediately by going to Dr. Seshikaran, just to set the stage, if I can. Dr. Seshikaran, can you just set the stage for the audience here in this panel discussion as to how the gut microbiome is involved in chronic systemic inflammation, and if there is a role for probiotics in mitigation of chronic systemic inflammation? Over to you.
Thank you, Professor Anura, and thank you, the organizers, for inviting me in this panel discussion. I think a lot has been said on the issue of the role of the gut microbiome as well as probiotics in systemic inflammation in the last 2 days in the various discussions. Basically, the mucosal barrier is maintained by the gut microbiome. And whenever there is a breach in the mucosal barrier due to dysbiosis, there is exposure of the immune system, which is just under the submucosal plane, to various gram-negative bacterial cell wall lipopolysaccharides. So the immune system then thinks that there has been an invasion by bacteria, and therefore it mounts an inflammatory response by releasing a whole lot of cytokines and other pro-inflammatory biomolecules. And now this ultimately doesn't have a target in the sense that there is no active infection at the moment, and therefore these inflammatory cytokines target the body's own tissues bit by bit. And that contributes to quite a bit of systemic inflammation associated various diseases. Now, if this were to happen repeatedly because of changed lifestyle or due to gut dysbiosis, this chronic inflammation, as described, becomes chronic, and it has its own effect in the form of causing metabolic syndrome, non-alcoholic fatty liver disease, damaging the endothelium, leading to cardiovascular complications, so on and so forth. And as we saw in the recent COVID pandemic, that those who had heightened chronic inflammation had a greater morbidity and mortality. The next part of the question, of course, is whether probiotics can mitigate this. This was also discussed yesterday, that there are certain specific strains of probiotics which have this anti-inflammatory kind of properties. And these probably will restore the mucosal barrier and prevent the lipopolysaccharide constantly stimulating the immune system and thereby toning down the chronic inflammation. So we need to actually have a lot more research in this area. But certainly one thing will help is that if we have a proper diet, a proper lifestyle, we will automatically have a good microbiome, which in turn will reduce the chronic inflammatory load. Thank you, sir.
Thank you, Dr. Seshikaran. And of course, I am remiss in, of course, that Dr. Ganguly is a looms large in this conference. He is everything to everyone. And sometimes you forget to introduce who is everything to everyone. I'm sorry about that, but let me tell you that Professor Ganguly, who needs no introduction, very briefly, he is the former DG of the ICMR, former director of PGI Chandigarh and National Institute of Biologicals. He is a former president of the INAMS, Indian Science Congress, JIPMA, Puducherry. He's everything to everyone, literally, in this. And he has published so much, 775 research papers, and over 100, a century of PhD students. That is amazing. He has been honored by just every award that there is, including the Padma Bhushan in the field of medicine. So, Professor Ganguly, I'm sorry I did not ask you first, but could you give your comments to set the stage for this panel?
Actually, the comments which I like to make is highlighting few major things which I saw today and yesterday. One of the major area which impressed me was Dr. Oishi's putting the friendly microbiota into actual persons, perfusion, through perfusion, and then measuring the physiological and immunological changes and benefit there. Then Jeff Gordon's work, which again was given to gnotobiotic mice and then given to 15 children in Mirpur to get appropriate signatures. And then Tahmid Ahmad's work, where he could give the complementary food, which he calls the RAF, and this is ready to use food, and has tremendous composition, but in which he is adding a particular microbiota combination which came from Jeff Gordon's work and is carrying out a very large study on enteropathies. But what was very endearing was that these guys have not only worked on the immune system, they have worked on the biomarkers and they work on the proteomic signatures and ultimately could correlate the metabolic pathways, functional pathways, and the benefits which could be measured very nicely. So this is something which I have never seen over here. And the papers were published in Journal Science, two papers are published in New England Journal of Medicine. So it is very good it is very nice that we could do this. So I will highlight this as one of the crowning glory in the science of the probiotics, which we saw today. Another aspect, two aspects which I want to highlight. One was Dr. Hachimura's work, where we could see actually that what's happening at the intestinal mucosal surfaces, where when you probe it with the lactobacilli, what benefits it provides and how the IgA and the immune system is modulated. This was again something which was actually you are seeing in the kaleidoscopic picture, that how lactobacilli are building up the immune system, including the germinal follicles. So this is something, as Dr. Pandai is sitting here, he's one of the major readout for any vaccine or anything which he uses, that whether it could produce a good germinal follicle. Dr. Hachimura showed this actually in a very nice person, functional. Last paper which actually impressed me was a real good clinical trial for the CIDs, the common infectious diseases. We always got stuck when we were looking at that whether this works or whether it doesn't work. So she did some very good meta-analysis, really good placebo-controlled trials in about 600 to 1,000 subjects, and there it was given for 12 weeks, although the severity and duration was not affected, but she got some very good up to 0.01% of statistical strength in incidence and prevalence. So it was a really very nicely protective. So I think these are some path-breaking things which is happening in the area, and it is going to benefit us tremendously in the future. And when you looked at the posters and others, they were so good science-driven work which is moving. I feel that we have a very good future for the scientists who are working in this area. Thank you, Dr. Kurupad.
Thank you, sir. I think that was a superb summary of what we have heard so far. So taking cues from you on inflammation and continuing with what Professor Shashikaran had said earlier, can I move to Professor Ramakrishna and ask him if he has any real world evidence that what Professor Shashikaran was saying is true, that manipulation of gut bacteria ameliorates infectious disease. Over to you, Rama. You're muted.
Yeah. So sorry. Yes. Thank you for that, and thank you to the organizers for asking me to be here. It's been an interesting 2 days. The quest for probiotics is more than a century old, and essentially what we want to do is to use this knowledge to change lives, to change our health status, to improve our health status, and to prevent disease. But do we really have the clinical evidence for this? What we heard yesterday from Shiv Pillai initially was about the pathogenesis of, in contrast to what happens with chronic diseases like Dr. Shashikaran was saying. Do probiotics really alter the course of infectious disease? Because that's really what we are considering in this particular symposium, in the 11th Probiotic Symposium. With infectious disease, the evidence is probably a little bit less than we have with chronic diseases. There is a lot of experimental understanding that probiotics alter mechanisms which can modulate infections, and particularly viral infections. Yesterday, Shiv also talked about the role of the commensal bacteria in changing COVID-19 pathogenicity. Subsequently, we heard from Dr. Bruno Port that about some studies which looked at viral infections and the immune response to that, and the probiotics and how they modulate that. So what we have eventually is that there are a lot of surrogate markers which tell us that probiotics may influence viral illness. For instance, we know that there are the plasma levels of various cytokines, and particularly of interferons, may go up to give probiotics. There have been multiple studies where influenza vaccines, and there are probably about 7 trials which looked at the effect of probiotics on increasing the immunogenicity of influenza vaccines. We know that when given to patients with viral infections, the quality of life is improved by probiotics. We know that viral titers have been shown in several studies to reduce with probiotics. There's a recent meta-analysis, in fact, just published, which looked at 27 clinical trials with approximately 9,400 patients with respiratory tract infections. And 9 of these used combination probiotics, 15 used single strain probiotics, and 3 used prebiotics along with probiotics. Essentially, what these show is that the symptoms definitely there is an improvement. in terms of reduction of severity of illness, as we heard Dr. Jensen say today in the last talk of the day, that she talked about how that particular strain of Lactobacillus casei in a meta-analysis showed that it improved outcomes, but it prevented illness, but wasn't so effective in curing an illness which is already acquired. So the role of probiotics currently seems to be in prevention. One of the largest trials, which actually is listed on clinicaltrials.gov, is a trial being conducted by Duke University, which is looking at 1,000 participants. And these are basically household contacts of COVID-19 patients. And the trial, I don't know what the status of the trial is currently. Essentially, what they are looking at is to see what happens to the microbiome, what happens to viral shedding in these participants. There are about 10 ongoing clinical trials on COVID-19. I suspect that by the end of the year, we will have the results of many of these. And since publication of COVID-19 trial results is accelerated, I'm sure we'll have the information to hand. So essentially, I think what we need is that many of the trials, there's a lot of heterogeneity because different strains have been used, different combinations have been used, the markers have been different. And so we should have something like what Jeff Gordon and Termid already told us, that you have one kind of study all around the world where everything is kind of conglomerated together. So you have maybe specific strain or a specific combination of strains that you use. We have similar markers in different settings and use a variety of measures to try and see whether you can get both laboratory and clinical markers put together. So we have great advances in our understanding of whether probiotics will help in attenuating viral illness once already we have achieved, we've got the illness. Thank you. Thanks, everyone.
Thank you, Rama. Just briefly, I want to thank you for actually putting two frameworks here. One of helping prevent infection or inflammation, and the second is actually improving the response to a vaccine, which are two different frames. And I think everyone needs to think of probiotics respectively in those areas. Just, I wanted to ask you back, in the Duke University study, is there any specific probiotic that you know of that is being used, that is being trialed?
So this is a combination of Lactobacilli and Bifidobacteria, which is being trialed in the Duke University study.
So it is the usual duo of Lactobacillus.
Yes.
Thank you very much. But at this point, can I move now to Professor Srivastav and ask him now to take it from here to understand the matrix in which probiotics should be given. Is there any additional advantage, Dr. Srivastav, of milk, of fermented milk, as a food matrix to give probiotics to human beings?
Oh, thank you very much. In a nutshell, we can say that in vitro characteristics attribute of probiotics may not be replicated exactly same in vivo if it is not administered properly. To administer the probiotics, either we can administer as sachets, as a capsule, or in a food matrix. This food may be fermented dairy, it may be non-dairy. If we talk about food matrix, fermented dairy products, yes, there are list of advantages if fermented dairy products are selected as a matrix for administration of the probiotics. There are 4 points which we should always consider when we talk about administration or ingestion of the probiotics. Number one, probiotics should sustain the acidic media of stomach. Number two, it should retain its genetics. It should retain its ability to colonize and to adsorb with the colon. Number 3, there should be some additive or synergistic effect of food in which it is added. And number 4th, which is most important, that it should not lose its pharmacological activities after addition when food is being processed. So these are the 4 attributes which probiotics should retain once it is mixed with the food. And out of these 4, at least 3 are confirmed if we are taking fermented dairy products. Because fermented dairy products or milk is a neutralizer of the acidic pH of the stomach. So survivability, sustainability of probiotic is enhanced if it is a fermented dairy products. Number 2, we found that there are several genes which are coding the probiotics. They are upregulated only during the process of fermentation. Otherwise, this pharmacological activity is not being seen. Number 3, which is more important, if we see the nutraceutical value or health attributes of the milk, milk is having lactose, which is converted into lactulose when we heat it, when we pasteurize it. And lactulose is a prebiotic. So it is a food for probiotics. So there is no need to add prebiotic when we are taking fermented dairy products as food matrix for probiotic administration. Milk is having lactoferrin. Milk is having galacto-oligosaccharide. Galacto-oligosaccharide is again prebiotic, food for probiotics. So it is helping probiotics to sustain, to multiply, to retain its pharmacological effects when it reaches to the colon. And most important, milk is having short chain fatty acid, butyric acid, which helps in multiplication of neuron, which we know that very slow multiplying cell. So if milk is used as a probiotic, so in addition to probiotics attributes, all additional effect is also seen. In addition to that, bioactive peptides, immunoglobulins, maybe IgG, IgA, all are present in the milk. So they are the not only additive, they are the synergistic. 1 + 1 = 3 if food matrix dairy, fermented dairy product is selected for probiotics. Nowadays in market, if you see, I tell you that 70% dairy products with probiotics are of nutraceuticals, are of food which we are using for our health attributes. So 70% are probiotics with dairy foods. So certainly, if we are using fermented dairy products, in addition to probiotics effect, we will also get synergistic effects of nutraceuticals of the fermented dairy food. So these are the clinical advantages if we are using fermented dairy products.
Thank you, Dr. Srivastava. That is very good response, which covered all the ground. I want to just now move to more specific issues, and that is to ask Professor Prajapati, is there any difference in the quality of fermented milk from different species of animals like cow, goat, or camel? And is there any role of bioactive peptides produced by probiotics in controlling COVID infection? So sorry, I'm asking you 2 questions, but if you could briefly answer both.
Yes. Thank you, Dr. Kurpat, and also thank the organizers of the conference for giving me this opportunity. Yes, since the onset of this COVID-19 came last year, we got the first report from Nature that what is the structure of the SARS-CoV-2, and then how to neutralize it and how to prevent its binding to the cell membrane. And in this case, we are talking about many benefits of fermented milks. As Professor Srivastava already highlighted the significance of dairy. Here I am trying to focus the significance specifically of the peptides which are being produced as a result of fermentation of milk by specific probiotic cultures. even though we do not have many studies now, but there was one study which published earlier this year in European Journal of Pharmacology, where the milk-derived proteins, beta-lactoglobulin, and its fermentation produces certain peptides. And these peptides have been found to nullify or inhibit the spike proteins of the virus. So the spike proteins, especially S and its allele of this S1 and S2, which they are binding to the cell membrane, the virus is able to infect. So to stop its infection, these kind of proteins are very useful. Especially we are talking about angiotensin converting enzymes. And initially the paper in Nature has indicated that angiotensin converting enzyme is providing one of the site for binding of the spike protein of the virus. So if we reduce that concentration, naturally the virus will not be able to infect this the cellular our cells. And for that, angiotensin converting enzymes, the peptides related for this, they are very useful. So we have a lot of studies now done on bioactive peptides from fermented milks. And as Professor Anura, you indicated, we have also done in our studies laboratory, these studies with different probiotics and using goat milk, cow milk, and even camel milk, and to study the pattern of different kinds of peptides. Even though we have not done any specific work for the inhibition of SARS-CoV-2, but indirectly if you correlate that if we have the peptides which are known to have angiotensin converting inhibition enzyme activity, and also the antioxidative potential of these peptides, this will definitely help in building not overall immunity, but also in fighting against viruses. So especially specific studies against virus are very limited, but there are plenty of studies which demonstrate the production of peptides from this thing. And the production of peptides depends upon the fermentation conditions, the type of probiotic culture used, and also the species of milk. And that is why we know that, say, if we have the milk from camel, it has got more of the peptides which gives anti-diabetic effect. We have seen probably latest work on donkey milk also, which has given a very good activity against the this insulin resistance and so. So the species of milk definitely relates to the type of peptide which it produces. However, the activity of production of different peptides is strain specific again. All cultures, all lactobacilli or bifidobacteria, they are not able to produce all types of peptides. This is also limited to its proteolytic activity, and this peptide production is also strain specific, as we have the strain specific effect of any other probiotic culture. So we need to screen the cultures for its ability to produce different kinds of peptides, and then we see the pharmacological effect of the peptides into this. So we have a series of studies on these things, and probably in coming a year or so, we'll be getting peptides packed into bottles for treatment of different kinds of infections and so. We have at the moment peptides available as a nutraceutical over the counter in the pharma stores also for boosting your immunity, also controlling your hypertension and diabetes as well as so. So I would like to inform to the house that we have a lot of potential, especially in fermenting milk, and that too the whey proteins, the beta-lactoglobulin and alpha-lactoglobulin, they are able to produce the peptides which are having specific physiological functions in the human body and so. So definitely we prefer camel milk for especially diabetic patients. We prefer goat milk for especially those who want to have very ease of digestion, because that we take it from childhood, that goat milk is easier to this. However, fermentation pattern is important, and the type of culture which you use for fermentation is very important. So I hope I could justify both our questions. Dr. Kurpal, please.
Thank you, Dr. Prajapati. I think one of the key issues is fermentation and the generation of these peptides in a milk medium outside the body versus fermentation and the generation of bioactive peptides within the colon or the lower reaches of the small intestine where they may have different activities. We'll come back to this, of course. But right now, I just want to tell whoever's watching that there is a chat box in which you can type your questions. I already see there are 2 questions. I'll come to you, but I just want to first engage with Dr. Anita Jatana from Apollo Hospitals. Dr. Jatana, can you tell us on the ground what the use of probiotics has been in your experience and how has that helped? Have you had any experience of this with patients who have been admitted with COVID, for example, or any other disease, in fact?
Well, I'll just first go back to maybe a couple of decades back when we heard this first word probiotics, and with the same sentence we were told, well, it's not still available in India. So in the meanwhile, keep taking curds. But soon we had Dr. Nirja there and she introduced it, and then there's been no looking back. I think I've attended all the conferences, and thanks to all the researchers who gave us a lot of knowledge, and armed with that knowledge, we used it in our hospitals. So I've had a lot of experience and very good experiences. Initially was a little apprehensive because, you know, the patients are in sepsis, they are in shock, you don't know you should do it or not. But we used it first for the antibiotic diarrheas and we found excellent results. You know how patients, when they are there, it's a battery of tests and a battery of maybe, you know, literally a handful of antibiotics and medicines they are on. So it definitely helped us a lot with those patients when they were there. We replaced the juice which was going to our patients at mid 11 o'clock, and we replaced it with a probiotic drink. And we found they did much better in their, you know, tolerance to medications. And supported with a lot of science and research, which we've been attending to all these conferences, we moved forward to celiac and we found good results there. Not to say that they can take wheat and then take probiotics. No, they have to eliminate the wheat and the wheat products and the gluten. But we found that they fared better in their symptoms and their growth and their diarrheas, the IBS, the IBDs, the constipation. So it was not only diarrhea that we found good results, we also found good results in constipation. So it's actually part of our indoor work that we do, and also forms part of a lot of counselings that we have in the OPD, where we tell them to use probiotics, because maintaining a good immunity and maintaining a good gut, we all know, is of huge importance. So supported with all that, we've really gone ahead and used it a lot. For COVID, we've not been been able to give you any studies, but the others, as I said, we've got a lot of literature that we've got good results with the other patients. But for COVID, I think we'll have to wait for a little more to see that. We did give it to them while they were there, definitely, because again, the gut was compromised. But I don't have literature to say or data to tell me how well they benefited from that. But in the others, we've got a lot of support, supporting data and good results. And I'm absolutely very confident using it in all the patients. So what I hear here from all the researchers, we take it back to the hospital scenario and we use it with good results.
Thank you. Thank you. Let me just read out a couple of the questions that are there on the comments panel. The first is to Dr. Seshikaran, which is that it is from Ashok Vaidya, and it is that anti-inflammatory rasayanas with polyphenols, anthocyanins, etc., positively influence gut health. Do they have any influence on the gut microbiome? Dr. Seshikaran, and after you,
I'm not too sure whether there has been any studies directly on these, but we can assume that many of these polyphenols also act as prebiotics, and they will probably specifically promote the growth of some of the beneficial bacteria and thereby help in reducing the inflammatory load, and thus having the effect. They will therefore have a benefit of both the the polyphenol per se having its antioxidant effect, and also polyphenol acting through the gut bacteria to generate the anti-inflammatory effect.
Actually, Dr. Korupad, if I could try to answer this. There is a scientist known as Dr. Bala Subramaniam. He was from Vellore. and he published a lot of paper on the gut environment free radicals which are generated, and was very worried that what these free radicals will have impact on damaging the gut mucosa as well as acting on the flora in the gut. Some of them are susceptible, those who do not have catalase or those who do not have good antioxidants, they might get damaged. So antioxidants may provide an environment within the gut which might favor the colonized habitat of some of the organisms. It also does one more thing, that there is a mucosal water interface in the gut, and ammonia, development of ammonia, those who are gastroenterologists here, they will appreciate it, is very sometimes very damaging, and this also creates a better interface over there. So these are some of the things, but Balasubramaniam's work is a major body of the work which is available in this area.
Thank you. Although I know Rama can answer partly these questions, I'm going to direct the next question to you, Rama, specifically. And that is that there is too much dietary fiber in traditional diets of Africa to not much in present American diets, and in between in Indian diets. So is there any relation of the geographic diet to diseases vis-a-vis the use of probiotics? This is a question I'd have asked Dennis Burkitt, but since he's not available, Rama, it'll have to be you.
That's a difficult question. Certainly, Burkitt was a champion of that hypothesis, and there's plenty of evidence for that. So fiber comes in different forms. There's different constituents of fiber can enhance the growth of different communities of bacteria. So what we had with the original Burkitt's hypothesis was that the African diet with the large consumption of fiber, of maize, etc., was protective against colon cancer, to protect against diverticular disease, and a variety of other kind of non-communicable diseases. With regard to probiotics, so what we are trying to do, or what people who want to intervene to change the way the microbiome or the structure and function of the microbiome towards a more healthy system, is to give prebiotics, which essentially could be fiber, components of fiber, and enhance the growth of selectively of certain bacteria, maybe the probiotic. So symbiotics certainly as interventions have helped. Whether the diet alone is responsible for differences in the incidence of disease in different parts of the world it's certainly likely. We know from migration studies that when Indians migrate to the US, we certainly are much more likely to develop inflammatory bowel disease. And these were studies, for instance, in which Jayanti has contributed a lot. First generation Indian migrants to the UK had a slightly higher incidence of Crohn's disease and ulcerative colitis. Their children, which are the second generation migrants, had an incidence of ulcerative colitis and Crohn's disease, which were higher than the Caucasian population. So clearly, there is an influence of environment. And this influence is probably because there is a change in diet, there's a change in hygiene, and this leads to a change in the microbiome. Now, many years ago, many decades ago, this was shown among the Japanese, colon cancer was very low. But when the Japanese migrants to Hawaii or Asian American migrants, people living in Hawaii, they've shown that the colon cancer goes up very rapidly. And this has clearly been related to diet and to bacteria. There are many such instances. And, specifically if we want to look at one chronic disease which Dr. Shashikaran was talking about, I think where we are facing problems will be with cardiovascular disease, with joint disease, and with inflammatory bowel disease and diabetes. Diabetes, I think South Asians are already shown to be extremely highly prone to develop diabetes. Whether probiotics will modulate the incidence of this is something I think that could remain a bit of a challenge.
Thank you. Dr. Shashikaran, do you have an opinion, or would you like to wait till I ask you a more provocative question?
No, I think I'll wait. Let us go on further.
All right. There's some more questions, and I think there's one question on the release of the BCM7 peptide from from A1 beta casein, and there's no one better on this panel to answer that question than Dr. Srivastav. Dr. Srivastav, what is the evidence of release of BCM7 peptide from A1 beta casein by fermentation in the human colon?
Okay, thank you very much for this question. At present, let me say that there is a big unrest in the society about beta A1 protein and beta casein A2 protein. I would like to clear the myth about A1 A2 protein and A1 milk and A2 milk, which is going on in our country. The first thing, the difference between A1 milk and A2 milk, what we say, is of only one amino acid. In A2 milk, at position number 67th, it is proline, and in A1 milk, at position number 67th, it is histidine. That's all. The story of A1, A2 started from New Zealand. In early 90s, scientists, a group of scientists, they found that children from some island staying in mainland, they are less susceptible to diabetes than the children of the island. And then they correlated that in mainland, they are consuming more milk, so they are more diabetic. And they found that in milk, the beta casein protein was A1. So they started doing the epidemiological study, and they say that, okay, A1 is responsible for diabetes, and later on A1 milk has been correlated with many diseases, schizophrenia, autism, many diseases have been correlated, arteriosclerosis, that is responsible for that. For India, the concern was that we are using the bull of exotic breed, Holstein Friesian, which is of the US, Europe, and New Zealand, for our crossbreeding program. So a lot of problems started in our society at government level, that we should stop crossbreeding program because they are giving A1 milk. We should go for our indigenous cow because it is giving the A2 milk. So let me clear. We had done a lot of research at NDRI and National Bureau of Animal Genetic and Resources. Our all indigenous cattle breeds, 50 breeds, they are A2 milk. Our all nondescript cattle breeds, they are A2. Buffalo milk, A2. Goat, sheep, camel, A2. So in nutshell, there is no cause of any concern to India as far as A1, A2 milk is concerned. Not only this, our all crossbred cows from where we are getting the milk, they are A2 milk. Breeding bulls of exotic nature, Holstein Friesian, Jersey, which we are bringing in the breeding policies, they are 99.99% A2 milk. So first thing I would like to clear this doubt, that let us not create the panic that A1, A2 milk, our all India, our milk present in India, either from crossbred or from buffalo or from indigenous, they are all A2 milk. That is the one side story. Coming to the release of A1 and A2 peptides, A1 and A2 protein from beta casein, we all know that milk is having casein protein and whey protein. 80% is casein protein. There are 3 types of protein in casein: alpha, beta, and kappa. beta is having A1, A2, not only A1, A2, they are having 10 more proteins. What has been found till date is not proving anything that A1 is bad for health or A2 is good for health. Let it be very clear. We have reviewed all the literature. No confirmation that A1 is bad for health and A2 is good for health. Whatever report has been given or it is available in the literature that A2 is good for health, you will be surprised to know that all the research has been sponsored by A2 Corporation Company. All the scientists who have reported that A2 is good, they are the salaried employed of A2 Corporation of the New Zealand. Recently, this A2 Corporation has written a letter to New Zealand Food Safety and Standard Authority of India that the general milk, if it is not A2, means general milk should be given a word of caution that it can cause like this disease. Contrast to that, New Zealand Food Safety and Standard Authority of India, they have warned the A2 Corporation that they are giving the misleading advertisement and immediately they should remove this misleading advertisement that A2 is good for health.
Dr. Srivastava, can I interrupt you just a minute?
Please.
Because we have lots of questions. So I want you to come to critically telling us this A1, they are saying it's all a big myth, correct? A1, A2. And the release of BCM7 peptide, that will be there, right? From A1 casein, is that correct?
Yeah.
It will be there.
Yeah, you know, from A1, it is the BCM7, and from A2, it is the BCM9.
That's right.
That's absolutely correct. These 7 amino acids peptide from BCM, from A1. So they say that these 7 amino acid, they can cross easily through intestine, they can cross the blood-brain barrier, and they can act on opioid receptors, so they can cause adverse effects.
Correct. Thank you.
BCM9, they say that they cannot cross because it is 9 amino acid. But I tell you that experimentally, even A1 is also releasing 9 amino acid peptides, and BCM7 is also released by A2 milk. So there is no conclusive result that which bioactive peptides BCM7 is released only by A1 and BCM9 is released only by A2.
Thank you, Dr. Srivastava. It really have shown us the problems of conflict of interest in research.
And just to add that Government of India has already given a now clarification that there is no difference between A1 and A2.
Thank you.
FSSAI is given. Yes.
Now, I want to ask you, Dr. Prajapati, if you can answer this. Is there any work going on in India with regards to what the microbiome produces? Like they call the question refers to metabiotics. I suppose you could call it postbiotics or any product of the microbiome. Is there any work with that with reference to COVID going on in India now?
As far as I know, probably there is no specific work.
Does Dr. Fully know if there's anything going on?
No, only people try to check the efficacy of different peptides to see whether it has any effect on binding site or not. But with respect to other metabolites, there is not much work related to that.
Dr. Kurupad, there is a data for llama milk. Llama is related to camel. There is a published data. Llama milk has anti-COVID activity. So suddenly it got very popular and famous. But there is no data with the camel milk, but llama milk data is already published.
Thank you. I am going to come back to metabiotics in a bit, but I'll call it something else. Dr. Anita, there's a question about the use of probiotics with ulcerative colitis or colectomies. Do you feel that probiotics have a role if someone doesn't have a colon? And that'll go to Rama as well next.
Yeah, definitely. And then because they're going to be having a lot of lactose intolerance, so this definitely helps. we always, before the probiotics was available, only gave curds to them. The curd base was better tolerated. So this again definitely helps them in getting back to normal much faster. The number of stools are better controlled. So the gravity is much better controlled with this. It definitely has a role to that. The only place we avoid probiotics is sepsis in acute cases in ICU, but we've used it a lot extensively for all the diseases across in hospital.
Rama, do you want to give a very brief yes or no?
So yes, probiotics have been shown to be useful to induce and to maintain remission in mild ulcerative colitis. They are not useful in Crohn's disease. In the absence of the colon, in patients with colitis who had their colon removed, they can develop a condition called pouchitis, where probiotics have been shown to be useful.
Thank you, Rama. That was very, very concise. I just want to now end, because we're coming to the end of our time, to ask a provocative question. I suppose I would like you to attack me in response. And that is that I want to say, with all this talk we have of probiotics and prebiotics and all this, what we've heard of the MDCF and all that, eventually, let me ask you this question, that the ultimate action of these probiotics is through the production of certain peptides from their matrix, or it could be something that the bacteria themselves produce. At the end of the day, I want to ask, is it possible that we just cut to the chase? And if we had good enough science, stop all this and just get to giving people those postbiotics that they need. For example, if you feel they need short chain fatty acids, then you need to provide people with short chain fatty acids rather than worrying about them being generated. I know there are some startups, not in India, but abroad, that are beginning to look directly at what they call postbiotics, what others call metabiotics. And the whole question is cutting to the chase. Stop talking about the bacteria and the other things. What do you think? Can we start with Dr. Ganguly and then go through the panel? Should we continue to be talking about this sort of thing?
Actually, if we really look at the science of the probiotic, I don't know whether you listen to Dr. Pillai, Shiv Pillai's talk. There is so much in the food which we consume and the organisms which we have, the analysis of the huge amount of proteomic signatures and how they modulate our metabolic functions is not only limited to butyric acid or short chain fatty acids and other. There are huge number of peptides which actually and the gene signatures and transcriptomes which modulate our health. Particularly this was seen in the context of inflammation. And Shiv Pillai's data was on inflammation and how some of these things could affect the fibrosis of the gut, as fibrosis of the lung, as well as the lung inflammation. So the science is evolving over here. At the moment, we do not know what will happen. I will inject one small thing over here that normally, and this most of the gastroenterologists will know, normally gut doesn't allow proteins or peptides to enter. They normally go through our portal system, and if the Kupffer cells are in the liver are supposed to chew them up, otherwise we will have huge amount of antibodies to food and other microbes and other things. So what happens, one of the bad side of the milk is this, that this particular part I had researched with one of my colleagues, Dr. Lata Kumar, that lot of our children develop milk protein allergy, and milk protein allergy is manifested as uneasy tummy, some pain in the tummy, and children, when you look at them, they have developed antibodies to the peptides which no, the pieces 7 and 9, which Dr. Srivastava was talking about. So we have lot of malabsorption in children, and that is one of the major issue over here. Whether the intestinal barrier works for them or not, we do not know. So some of the things the probiotics do, they really create the mucosal integrity in a much better fashion that will prevent all of these. So the probiotics, in a sense, are very, very important to maintain our mucosal integrity. And that was also shown by some of the talks which were presented here with actual data set.
Thank you, sir. So you reject my my hypothesis, and that's fine. I want to know what Dr. Shashikaran.
I will not dare to reject your hypothesis. These are opinions, and and one doesn't know which opinion is actually will stand the scrutiny. So
Sure.
it is just an opinion.
Thank you. Thank you. Dr.
Hello,
Go ahead with the probiotics in the meanwhile.
Sorry?
We'll go ahead with the probiotics in the meanwhile.
Good. I was going to ask you. We'll come to you, Dr. Zetana. We're just going through each one again. Dr.
Dr. Kurpa, this question is always asked whether it's probiotic or whether it's nutrients. See, in the metabolic pathway, on one end you have the substrate, and the other end you have the bioactive substance. Should we give the substrate to the human being and make the human being make the bioactive substance, which is better regulated? Or should I directly give the bioactive substance? This is like whether should I fortify with nutrients or should I ask people to have a food-based approach? So the same question comes here too. Both have their place. I would vote always for the natural process and then keep the bioactive molecule only for certain special situations.
Dr. Sasikaran, you are true to NIN philosophy, the patriotic philosophy of food-based nutrition supplementation, no chemical. Or no, but I don't know whether I support that or not. I do not know. But NIN has a dogma, and Dr. Sasikaran actually, even though he's a modern NIN guy, but he carries that dogma.
Thank you, Dr. Shashikaran. Rama, can you step into this, into these flames now?
So I think the answer is sometimes yes and sometimes no, in the sense that, if you know that short chain fatty acids are going to be effective in a particular instance, for instance, in diversion colitis, it's the lack of the microbiota that leads to diversion colitis. But you know that if you just perfuse them with, you give them short chain fatty acid enema, that you can get rid of the diversion colitis. So there are specific conditions where you might use the metabolites. But for the most part, what we need to remember is that, as Dr. Ganguly was saying, that basically probiotics, the bacteria, intact bacteria, have a lot more. They are going into a complex ecosystem where there are metabolic interactions with different communities of bacteria. And an enzyme that is lacking in a probiotic may be present in another community. So when food is taken, or when something or a prebiotic is taken, it's not only the probiotic that works on it. If that probiotic lacks certain enzymes which requires this substance to be metabolized, the other bacteria provide that. And essentially this interaction, I believe, is very important in a number of the actions which we expect of a probiotic. And that's even for metabolism, even for colonic metabolism. And so I think more often we are likely to use the intact bacteria rather than the metabolite per se.
A quick riposte to you, if I'm just to keep the flames burning, is this: that if we feel that the bacteria are important, then are we doing the wrong thing in single bacterium probiotic preparations? Should we not be going for what might be called a cluster of bacteria which are perhaps better than the single bacterium approach? What do you think?
So, in terms of the evidence, sometimes you find that where we are looking at inflammation overall, a community of bacteria probably seems to be more, or a mixture, as you said, a cocktail of bacteria probably seems to be more effective. Now, we are talking here about changing disease. And so sometimes there is evidence that for ameliorating inflammation, perhaps a cocktail of bacteria might be more effective than single bacteria. But whether it works for all situations, I don't know.
I agree. I asked you a very controversial question. That's all. Thank you for attempting it. Dr. Ganguly wants to say something, or?
I wanted to say that these are like the sciences in the stem cells. There are pluripotent cells. One single bacteria might have several metabolic advantages, or a cocktail of bacteria could be designed to give several metabolic advantages. So VSL3 is one of the cocktails which has been very, very popular, and Lactobacillus casei LCS. We saw a lot of data on LCS, and we also saw one single data from Jensen today. So the question is still on, and unless we could do very good randomized controlled trials, we will not know the answers for the future. But I want to interject, like Bhavtosh is not sitting here. Bhavtosh presented a paper in which he found that Lactobacillus rhamnosus has 23 different signatures. So by the old literature or technique, you put up a Lactobacillus rhamnosus and may think that this is a uniform species, but it might have several Lactobacillus rhamnosus. And so with the modern techniques, we will get really some very good data sets with designer probiotic strain, which could be much better assessed than what we were assessing in the past.
No, I agree, sir. I think now that we hear that twins are not really twins, I suppose this is inevitable that we're going to see that the signatures of what we think as one species are going to be very different. Dr. Srivastav, do you have anything to add to this debate? Are you a person who believes that we should take something that we derive from milk, or should we drink the milk and hope that it is derived inside our body?
No, as far as milk is concerned, peptide intolerance, protein intolerance, lactose intolerance, it is reported, no doubt in that way, as Dr. Ganguly said, especially to northeastern region, lactose intolerance is very high. That is number one. Number two, what I will feel that both way, when we are using milk as a food, I will like that let nutraceuticals be isolated from milk when we use milk as a food. So that is the one aspect, that we will give the milk and let the biomolecules, pharmacological biomolecules, be isolated, or they may be digested, they may be separated from the milk, and it should go in the body and it should act. But on the other hand, certainly, I will like that bioactive peptides, the way their therapeutic use, their pharmacological activities is being established, I'm sure that in coming 20 years, there will be a tough competition between food industry and pharma industry as far as milk is concerned. They will be isolating bioactive peptides and they will be using in therapeutics. Strongest aphrodisiacs has been isolated from donkey milk. Strongest analgesic, antipyretic, anti-inflammatory, 67-fold higher than morphine agonist have been isolated from the camel milk. So the way recently, sir, one bioactive peptides have been isolated, which enhances the penetration of isoniazide in cell, intracellularly penetration has enhanced. So it will help in the treatment of the tuberculosis. So I will go in both ways. I will like that pharma companies should isolate some biomolecules. Food industry should explore the full potential of the milk and milk products as a food because of their nutraceutical value.
So much. You speak with so much passion.
Dr. Kurupad,
Yes, sir.
last interjection. Today we heard from Tamid, and we know that that the children to get over their stunting, as well as to have a linear growth and brain saving of the brain and bone growth, 150 milliliter of milk and 1 egg, and some complementary substances in the food. So I want to end with this message. I will talk no longer, because if you give me opportunity, I will go on up to 6 o'clock in the evening. So I stop here.
Oh yes,
1 minute, this thing before the industry comes up with all the variants. Anybody who's having lactose intolerance, taking curd helps, or taking paneer definitely helps. And if you cook it with the cereal, then the lactose intolerance is better tolerated. So you can change the form of milks, because the lactose release is slow if you've taken it or you've cooked it into a custard. So doing that till we wait for the people to minus the lactose problems for us.
Let me just finish with the, because in all fairness, I have to give him a word. Although I just do want to say that once Dr. Shashikaran described Dr. Srivastav to me thus, he said he's so enthusiastic about milk that he's probably the only man in the world who can sell milk to a cow. So thank you, Dr. Shashikaran. Dr. Prajapati, any final words here, please?
Yes, sir. I just believe that postbiotics or the metabolites have a specific purpose and it can be used as a pharma product. But if you want to have overall health and so many other things, you have to have natural flora enhanced by probiotics. So consumption of probiotic bacteria through milk-based products or any other means, definitely I will also prefer to be milk, so that we get multiple benefits of so many things. Because whatever effect we get is not because of a specific compound. It is a consortium of several compounds which are acting, and we take benefit of such things when we consume live probiotic to that. But no doubt, these specific isolates have their specific purpose and specific use. It will come in pharma industry, definitely.
Thank you.
So my preference is consumption of fermented milk, probiotic through fermented milks or other products, food.
It seems like the consensus is until we know more, we should be continuing with what our grandmothers told us was a good diet. And I think that's good advice. I'd probably follow it myself. And I think that we have to watch and see what these post-biome producers will actually come up with in the future. But thank you very much. It's been a wonderful panel. There have been many questions in the chat box, which we could not take up. But if Neerja could forward those to the panel, and I hope the panel will be willing to answer those questions for the people who asked them. Thank you very much. Over to you, Neerja.
Yeah. So ladies and gentlemen, requesting for the felicitation, requesting Professor N.K. Ganguly to kindly come on the stage, and requesting Dr. Anita ma'am to kindly come on the stage. Everybody please put your hands together. Everybody please put your hands together. So before we proceed, we have two main announcemements to be made. So you all have received feedbacks forms in the kit bag. Please fill them and submit at the registration counter outside. And one more announcement, all those who have joined virtually, they can download e-certificate. So now we have to proceed towards the next session. So we now come to the close of the symposium with the valedictory session. So for this, may I invite Dr. M.S. Chauhan, Director, National Dairy Research Institute, Karnal, India, to chair the valedictory session. So everybody, please put your hands together. Dr. Chauhan has been the principal investigator of 11 externally funded projects and co-PI of 9 interdisciplinary and interinstitutional externally funded projects. Besides research, he has also contributed in teaching and developed 4 courses of animal physiology and animal biotechnology for the postgraduate students. So Dr. Chauhan is a fellow of National Academy of Agricultural Sciences, fellow of National Academy of Dairy Sciences, fellow Society of Extension Education. Dr. Chauhan has published over 155 original research papers and cited 7 books. 44 scientific and technical publications have been authored or co-authored. 34 lectures delivered in national and international conferences. Everybody, please put your hands together. Over to Dr. M.S. Chauhan.
So whether this speaker will be online? Okay, thank you so much. Okay, thank you very much. And at the outset, I would like to thank Professor Ganguly and Dr. Nirja Hazela for giving me this opportunity to be here in this last session. And in fact, it was wonderful session in the morning and then also the yesterday evening. And I have been given this task that the speaker is a civil servant here, Dr. Miss Anushi Sharma, and she's a Director, Social and Behavioral Change, Food Safety of Standard Authority of India, FSSAI, New Delhi. Miss Anushi Sharma, Director, Social and Behavioral Change and Food Fortification Resource Center, in a civil servant as an officer of Indian Revenue Service. Her current assignment at FSSAI involves leading the Eat Right India initiative and extending support to scaling up of fortification across the country. She has previous work experience as a director, National Health Mission in Haryana, where she was looking after the supply chain management of drugs and equipment and adolescent health pertaining to menstrual hygiene and weekly ionic folic acid supplementations. She has an interest in the area of public policy, administration, and social sector. In fact, I was fortunate to have FAD 19 dealing with this, the food safety aspects in dairy science. And we had an interaction with the FSSAI many times. And very recently, we were able to submit 3-4 protocol established by our NDRI to FSSAI, which was subsequently implemented by them. So I'm fortunate here today, Ms. Sharma here. So then over to you.
Thank you so much. And I would like to thank the Gut Microbiota and the Probiotic Foundation for inviting me here today. It's really heartening, to listen to the panel discussion which was going on before and to see this interest which is coming across people regarding improving the gut and healthy diets and habits which can be taken so that, we no longer have disease to start off with. If we can take care of our health, we don't need to take medicines. So that's one of the reasons why the initiative called the Eat Right Initiative Eat Right India Initiative was launched by FSSAI. And I would like to share a short presentation, in fact, through which I would like to take you all through the Eat Right India program. and why we started off with it and its importance really. So the vision of this particular program was primarily to provide plant-based food for everyone, which is also sustainable in the long run, and it is based on our traditional food culture. Why this becomes very important is because while we are having this great discussion about probiotics, what's really important is also to realize that traditionally we've been having a lot of fermented and probiotic food naturally. And somehow it is getting lost because the younger generation needs to realize the importance of cooking and getting and retaining that knowledge. Why this initiative of Eat Right really came about was because if we look at the Indian situation today, we've got almost 1.35 billion people. And a lot of people are dependent on the public distribution system for their requirements of carbohydrates primarily. With that, we also have about 4.3 million, and now, of course, the figure's gone up. I think it's about 4.9 million food businesses which are licensed. But a large number of the food sector in our countries and also in the unorganized sector. Now, what that really means is that a lot of people in our country end up having foodborne illnesses. There's also micronutrient deficiency. There's the growing concern of NCDs. So you have on one side unsafe food. You've got unhealthy diets, which people are adopting. And it is also overall leading to environmental degradation. Now, when we see all these things, what really comes out is no matter how healthy you are eating, if it is not prepared in a hygienic condition and you fall ill, you will not be able to absorb the micronutrients. Or if you are having fast food or you are having foods which are high in salt, fat, and sugar, you will end up with an NCD which will, in the long run, affect your health. So what really becomes important is that how do we promote through regulations, through facilitation, a system in which the food ecosystem of the country becomes safe, healthy, and sustainable. So FSSAI's mandate primarily is, of course, creating standards. We've got a vast number of labs where food testing is done. We've got the regulation and the compliance to ensure that rules are followed. And we've got extensive training and capacity building. But what we also realize with this mandate is that once the food reaches the citizens, how much are they aware about the importance of having home-cooked meal, which is prepared in safe and hygienic conditions? Because once the food has left the farm, it goes through the stages of processing, storage, distribution, retail, preparation, before it finally comes to us. So therefore, FSSAI moves ahead from being just the regulator to the enabler. And the Eat Right India movement, therefore, really wants to promote eat safe, eat healthy, and eat sustainable. Eat safe, like I've been saying, means personal and surrounding hygiene, hygiene in the entire food chain and combating adulteration and eliminating toxic residues. Healthy means having a balanced diet, eating less, eating on time, having a diet diversification, eliminating trans fat, reducing salt, fat, and sugar, and eating fortified staples. And sustainable means eating local seasonal food. A lot of fad happens where people want to eat things which have come from long distances, or things which have come from abroad are necessarily good. No, that's not true. We have in India a number of superfoods which provide us with enough nutrients and which are also sustainable. So sustainability also comes when we encourage people not to waste food, recycle plastic, and conserve water in the food chain. So what we do really is have a whole of government approach on the one side, where we try to link with other ministries and departments so that everybody's mandate when it comes to food has a common message. So we have been having a lot of interministerial discussions, like with the Department of Public Distribution, with food processing, with environment, agriculture, so that we have programs where there is a synchronization of messaging. On the other side, we have the whole of society approach. No program of the government can go just by making rules or by making laws. For the implementation, we need professionals, we need industry, we need development agencies to work with us. Forums like this, where discussions are going on, They serve purpose in telling people the importance of having a good health and a good gut. So some of the things that we are really looking forward is how we can have new standards. One of the things, areas that we are working on is the front of the pack labeling so that we can identify foods which are high in salt, fat, and sugar. We've already come out with regulations for reducing trans fat. In fact, by 2022, 1st January, only 2% of trans fat will be permissible. As of now, it is 3%. We've already got standards for fortified staples in place like rice, wheat, salt, milk, and oil, even for processed food. We are working on other staples also, which can be fortified. We are also working on strengthening our laboratories. We are working on training and capacity building of the frontline health workers, as well as of the food handlers in the organized and the unorganized sector. Benchmarking and certification programs are going on for street food vendors, fruit and vegetable markets, hygiene rating of individual outlets, for which we are working with the e-commerce platforms such as Zomato, Swiggy, etc. And we are trying to make sure that the restaurants on their platforms are also hygiene rated. We are also working with schools, with various institutions, so that they become Eat Right campuses. What it really means is that at early age, children become aware about the importance of eating well. So we also came out with a regulation which bans the sale of high fat, salt, sugar items in schools. This we came out in July 2020. We are also working with the food businesses to voluntarily adopt reduction of fat, salt, and sugar, reducing food wastage and promoting food donation, and adopting alternative packaging material from plastic. To create social behavioral change, we have the food safety on wheels, which moves around in various parts of city and tells people how they can check adulteration, or if they have certain issues about food safety, they provide guidance. We also plan to have a number of Eat Right Melas in the later part of the year, where people will not only be able to enjoy local delicacies, but also witness what the Eat Right India movement is all about. And we also hope to now use social media in a big way. Ever since the lockdown, in fact, social media has come in a big way to encourage influencers, bloggers, and have posts which encourage people to eat healthy. We had a number of books which we came out during the COVID lockdown about the importance of eating right to make ourselves strong, so that it's do you eat right? A lot of us have misconceptions about what eating right really entails. So that was another thing we came out. And for canteens and for places like restaurants and hotels, what are the guidelines that need to be followed during and post-COVID? We've also come out with that. So what are the broad 5 key outcomes that we've really talked about through the Eat Right initiative is how we can reduce the burden of foodborne illness in the country, lessen micronutrient deficiencies, reverse the burden of preventable diet-related NCDs, reduce carbon footprint of the food sector, and ensure a more sustainable food sector. And why all this is important is, like we've been discussing today, is it is the health that is the true wealth and not pieces of gold and silver, as Mahatma Gandhi had really said that. So thank you so much for your patience. And I hope you enjoyed hearing about the Eat Right India initiative of FSSAI. Thank you so much.
So it's over, or any questions? Any question? Okay. Anyway, thank you, Ms. Sarma. Professor Ganguly is here. Just I was wondering that, can we have with food like iodine we have in salt? Can we have a probiotics in every, like the basic food, where we can have a so that will be defined.
the way today. Actually, the food uh, modulates the microbiota. There was Gates Foundation funded a lot of studies, basic studies, where probiotic animals, that is germ-free animals, were used. And when you gave the food, and with the food they didn't remain germ-free. Gradually they got various microbial population, or you gave to these animals microbial population a particular food, these could get into these. So one of the best things which has happened now, that although we were having various probiotic, food-based probiotics, but now the food, ready-to-use food, which is given to children, Tamit's work has shown that we could distribute it at a very large scale with definitive benefits. Yogesh also did a very good study in Pune, where he gave the chickpeas with probiotics to again promote linear growth in the children. And so there could be food, some of them so-called probiotic food is available in market, but they might not qualify to be probiotic food, but science-based supplementation of food products is the future, and it will definitely come up. But we need to really have some kind of thinking on this. Like if we think that with the probiotic strains we will provide folic acid or vitamin B12, we need to think that whether it could be sufficiently provided or not, because India has got the largest population of spinal cord defect and as a disability and death because of the folic acid. We didn't understand the folic acid supplementation or fortification well. In UK, you cannot buy a bread without folic acid, and we didn't understand the sufficiency and deficiency, so we didn't use enough folic acid. So whenever we are putting probiotics and we think some properties will go there, we need to see that whether it is enough or not. Thank you.
Okay, thank you, Ben. One small out of the box question. I heard from you 3-4 times stem cells, pluripotent stem cells, and so can it be possible that through stem cells, if you insert the probiotics inside the stem cells and subsequently multiply them and have a small capsule, okay, this is the stem cell capsule, if you eat this, it will maintain your gut.
I'm ignorant about this particular part. I quoted stem cells because I thought that there will be some bacterial strains which will have a multiple effect. So
exactly, yeah.
I quoted pluripotent stem cells on that context, but I have no idea whether we could deliver probiotics through that. It will be very expensive.
Yeah, thank you so much, and thank you, Dr. Nirja Hajela, for giving me this opportunity. Thank you, all of you, for listening patiently. Thank you. Thank you.
Well, thank you so much, sir. So finally, all the good things come to an end, and this 2-day scientific feast comes to an end too. So ladies and gentlemen, we hope you all enjoyed the 11th India Probiotic Symposium, and we'll meet you again next year for the next in the series 12th India Probiotic Symposium. So ladies and gentlemen, I would now like to invite Dr. Nirja Hajela to give the vote of thanks. Dr. Nirja Hajela is the General Secretary of the Foundation, a PhD in biotechnology. She comes with more than 17 years of experience. Ladies and gentlemen, please put your hands together.
Thank you, Naina. Thank you. You did a great job. So thank you on behalf of the Gut Microbiota and Probiotic Science Foundation India.
It's my privilege and honor to be proposing the vote of thanks. So thank you for me is just not a word, but a million emotions of appreciation and gratitude. We would like to thank our visionary and mentor, Professor N.K. Ganguly, for his wisdom for establishing a foundation to propagate science that would enhance our knowledge and define our very existence. Our vice presidents, Dr. B. Shashikaran and Professor G.B. Nair, have been a constant support guiding us. Our governing body members, Professor B.S. Ramakrishna, Professor Anurag Kurpad, Professor Anil Kumar Srivastav, Professor J.B. Prajapati, Professor Ajay Bhalla, Professor S.K. Mittal, Professor Kia Lahiri, and Professor Tamang have always supported the foundation and have raised our bar for excellence. We are missing them today because they are not here with us physically, but nonetheless, we really appreciate their contribution. The encouragement and support from our senior members from Japan, France, and India, Mr. Shinji Hashimoto, Dr. Sarah Thompson, Dr. Junji Fujimoto, Mr. Tomoyuki Iwama, Mr. Minoru Shimada, is just overwhelming. They have played a pivotal role in helping the foundation achieve its objective. We would like to thank Dr. Samuel Raj, Dr. Ramendra Pandey, and SRM University, Sonipat, for so graciously co-hosting the symposium with us. Their effort and support in organizing the symposium has been invaluable, and we look forward to a long and continued association. We would like to thank our distinguished chief guest, Professor Shiv Pillai, and international and national speakers, Professor Tehmeed Ahmed, Professor Jeffrey Gordon, Professor Satoshi Hachimura, Professor Bruno Pott, Dr. Stephanie Jensen, Dr. Kenji Oishi, Ms. Inoshi Sharma for their excellent and very comprehensive talks. We thank you for making the time and sharing your thoughts on such an important subject. We would like to thank our chairpersons and co-chairpersons, Professor G.B. Nair, Dr. M.S. Chauhan, Dr. Amulya Panda, Dr. Saurabh Datta, and Dr. Amit Avasthi for so gracefully conducting various sessions and for a concise wrap-up. Thank you ever so much for your time. We would also like to thank Professor Anurag Kurpad for brilliantly moderating the panel discussion, and all our panel members, Professor N.K. Ganguly, Professor A.K. Srivastav, Dr. B. Sasikaran, Professor J.B. Prajapati, Professor B.S. Ramakrishna, and Mrs. Anita Jatana for sharing their thoughts and questions that keep arising time and again in most of our discussions. The panel discussion had helped clear many of our doubts, and I'm sure the audience is more than satisfied. We would like to thank all the young investigators for their enthusiasm in sending the abstracts. We received 60 abstracts this year, and the abstracts were so good, excellent, that it became difficult for the governing body to evaluate them. We would like all the young investigators to know that each one of you is a winner, and would also like to congratulate you for the excellent research that you all are pursuing in quest of improving public health. We would like to applaud all of you and each one of you for your efforts. So I think a big round of applause for all of them. Thank you to the event agency, Elborn Conferences and Private Limited, led by Mr. Rajiv Pandey. Rajiv, where are you? Be nice if you just stand up. Is he here? In there. Who holds the torch and ensures that all our events are a great success. This has been a very challenging and difficult year with the pandemic, which has entailed a lot of modifications and improvisation. So we just have about 30 delegates who are here physically, whereas last year for the 10th India Probiotics Symposium, we had more than 400 delegates. So there has been a change. And a very, very special thanks to Ragini, to Vishal, Karan, Shashank, and Ruhani, who took my call no matter what time of the day, for their dedication and for organizing this event so meticulously. A big, big, big thank you. A special thanks to Preeti. Preeti, I think you're there. Anushka and Priyanka for all their efforts. That's my team. And an interaction becomes only meaningful with an enthusiastic audience. We were overwhelmed with your questions and active participation. And last but not the least, a heartfelt thank you to our delegates both who were with us physically and virtually. So we had more than 800 delegates yesterday who were online, and today more than 600 delegates who have been online. So thank you very much for your time. This symposium would never have happened without your unstinting support. We thank you all for being with us on this weekend and making the event so successful. We hope you enjoyed the 2 days of scientific deliberations and look forward to meeting all of you physically in the 12th India Probiotics Symposium. So thank you very much. And I request you, thank you. Thank you. Yes, and for finding Naina for. She did an excellent job. Thank you, Naina. And so I just request all of you to join me for the national anthem. And with that, we close the day. Thank you. Thank you very much. Thank you.